So good morning, good afternoon, good evening, everybody, wherever you are in the world following this 2023 Rising Stars of Light event. We have a fantastic lineup of speakers today for the faculty track. But before, let me introduce myself. My name is Giulia Tagliabue, and I'm assistant professor at EPFL, and I will be chairing this event today. And then let me introduce you our committee members. Today we have the faculty track. Our committee members today are Lan Yang from Washington University, St. Louis. We have Andrea Alou from CUNY. We have Aida Nozgan from UCLA. We have Professor Harry Atwater from Caltech. We have Professor Ming Chu from Westlake University, and we have Professor Dragomir Neshev from ANU, Australia. I think I want to welcome you all, and without any further ado, we will start with our first speaker, Francisco Robles from Georgia Institute of Technology, that is gonna present us his work on accessible label-free optical detection. Sorry, I lost the screen. Optical microscopy with quantitative structural, molecular, and functional contrast. So the floor is yours, Francisco.
Can you guys hear me?
Now we do.
Okay.
Yeah.
All right. So let me try that again then. So my name is Paco Robles. I'm an associate professor at Georgia Tech and Emory University. And today I'd like to tell you a little bit about my lab's latest efforts in developing accessible, low-cost, label-free optical imaging technologies that deliver quantitative structural, molecular, and functional contrast. And in fact, all the images that you see in this title slide were developed with such system. Before I go any further, though, I do want to acknowledge everybody who has made this possible, in particular the members of my lab who are extremely dedicated to this work, our collaborators across Georgia Tech and Emory University, across the US and internationally as well as their funding sources that make all this happen. Okay, so first I would like to walk you through some of my background to understand how we got here. So throughout my career, my work has focused on leveraging different types of light matter interactions to enable endogenous molecular and functional imaging. And I broadly like to put this into two buckets, two categories. You have linear interactions and nonlinear interactions. With the linear interactions, you have things like absorption, scattering, and dispersion, which is related to absorption. And generally speaking, these are relatively simple technologies. They're relatively low cost, especially compared to nonlinear technologies. However, they do have limited label-free molecular information. If you're looking at absorption, for example, your primary target is going to be hemoglobin, maybe melanin, but that's kind of boring in the linear regime. You also have limited subcellular detail in 3 dimensions. You do have OCT and confocal reflectance, but the amount of subcellular detail that you get from this technology pales in comparison to what you can do in transmission mode. So there's some limitations there. Now, these limitations can be addressed with nonlinear technologies. They provide many more types of interactions that really provide rich molecular label-free information. They also provide unprecedented subcellular detail in 3 dimensions. However, they have the opposite problem. They tend to be complex and really expensive. And I'd like to dive a little bit deeper into this issue. So take one of the systems that I used to work on during my postdoctoral training. This is a pump rope system. The details of this are not really important. However, the system itself is comprised of a couple of modules. You have the laser scanning system, which tends to be a little bulky and relatively complex. This is representative of what you would get. And that delivers really beautiful images. But in order to get these images, you have to have this additional piece back here, which you often don't see the actual optical components. This is comprised of a lot of optics. You have a lot of different pieces like prism compressors, delay lines, and so forth. And all this requires a lot of expertise, a lot of time to put together, and it's expensive and complex. And really, this really motivates the work. And as I started developing my lab, I started asking, can similar information be obtained with a much more compact and simple and inexpensive system? Yes, there's going to be trade-offs, but can we start bridging the gap between what these nonlinear technologies can do and what we can do with simpler linear technologies? So we started addressing this problem, and we started first with a very old technology that has seen a resurgence in the past few years. We started with deep UV microscopy, which enables extremely rich label-free molecular information. Deep UV microscopy is not new. It was actually first pioneered by August Scholar, one of the founding fathers of microscopy, back in 1904. However, the way in which you detected the light, and the light sources were not really great. So this never really went anywhere. There was a second wave in the 50s, but by then you already had phase contrast. And given the complications that still remain in the light sources and detectors, it never went anywhere again. It wasn't really until about 2007 when what I call the third wave of UV microscopy came along. And it was really introduced by this paper by Matsueda at MIT, where they showed that you can quantify nucleic acids and protein mass down to the femtograms by simply illuminating the sample with 260 and 280 nanometer light, which is just amazing. Now, they also showed that you can image live cells for many hours if you can manage the UV exposure, which is also phenomenal. Now, there's obviously a lot of information in the deep UV region of the spectrum. It's not sharp peaks as you have in Raman and other regions of the spectrum, but there is a lot of molecular information. So my lab started playing around with this. First, we started with a hyperspectral approach. We started quantifying the absorption and the refractive index and plotting these complex plots where you have the absorption refractive index do these little loops when you have resonances. And this is just to show that there's a whole lot of information here. We were able to do phase maps and all these cool stuff with this interferometric approach. But as we were moving forward with this technology, we really wanted to make it as simple and as bare bones as humanly possible. And this even was too complicated. So we said, okay, let's strip all the complexity and just simply do transmission-based absorption imaging, where you can have a broadband light source and UV filters, and then just a microscope, or a bunch of LED light sources, and then just a microscope in key wavelength regions. And by the way, this paper was just published this past year, where we show that you can make these systems with very precise 3D stages for less than $5,000. So this is compact and low cost. And so now we're going to use the system and apply it to a bunch of different applications in biomedicine. The first one is going to be in hematology, where we found that by simply illuminating blood cells with 260 nanometers, 280 nanometers, and 300 nanometers, and weighting those three attenuation maps differently, we can recapitulate the gold standard stains for hematology, which are these Giemsa stains. And the agreement is just remarkable given the simplicity of UV microscopy. So we can differentiate all different types of white blood cells, the three different types of granulocytes, lymphocytes, monocytes. We can look at platelets, nucleated red blood cells, sickle red blood cells. We can quantify hemoglobin, and we can even look at bone marrow aspirates, which is what you see in here, with an extremely simple device. So now you can start imagining taking these very expensive and complex hematological analyzers that require a bunch of chemical reagents, daily calibrations, and making this into a very simple bright field microscope. And this is the same system to scale with this bulkier device. And so my lab has been using advancing this to provide passive microfluidics, enable pipelines for automated segmentation and classification. And with that, we've shown that you can do a 5-part white blood cell differential. And we're targeting applications like neutropenia, cancer detection, and identifying spicules in bones. We can even provide clinical images that look just like what clinicians are used to looking at with a single UV illumination light. So this is fast and efficient. So there's a lot of applications with this very simple device in hematology alone. But now I'd like to switch over to another application of UV microscopy using exactly the same approach. Here, we're going to be looking at histopathology. What you're looking in the background here is a prostate cancer tissue section stained with hematoxin and eosin. And this is a Gleason grade 4 with inflammation here in the center. And what we can do is take an adjacent section that has not been stained, so completely label-free, put it into a UV microscope, and this is the type of information that you get. In this particular image, we're actually rendering the multispectral molecular information with deep UV microscopy into these colors that recapitulate the structure of a tissue. This bright yellow color is the stroma. The ruby red colors that you see right here are from the inflammation, and different glands take up this different shades of red, signifying that they have different molecular composition. And the resolution here is in the order of 250 nanometers in a simple wide field configuration. So acquiring millimeters worth of data only takes a few seconds. So this is incredible. And we can slice and dice the data whichever way we want to provide the best representation possible. So we can do multiple optical stains. Here, just color coding different molecules in different colors, or we can even do a diagnostic stain when we actually take the glands and color code them for benign in one color and malignant into another color. So there's just so much that you can unpack here with this very simple technology. We can also provide clinicians with images that they're already used to looking at. So we can provide them with virtual H&E images, translating these UV images to virtual H&E. This takes a little bit of AI. We can also provide them with immunohistochemistry looking images. And this requires no AI whatsoever. This is just the endogenous molecular information. And this is P63, which identifies basal cells. We've even shown that you can improve upon the gold standard because sometimes the stain is not taken up by the tissue, but the molecular composition is always there. And our method is robust, even when the stains actually fail. So again, there's a lot to do here with UV microscopy. I think it's in its infancy, but hopefully I've been able to show you that you can really provide a lot of rich molecular label-free information with this very simple technology. All right, so now we're going to shift over to addressing this other limitation, which is limited subcellular 3D information. To do that, we need to talk about phase contrast. Phase contrast is arguably the most well-established manner for obtaining cellular and subcellular detail in cells. And if you can do it quantitatively, even better. So with quantitative phase imaging, what you're quantifying is actually the optical path length, which is the thickness and the refractive index. And this provides extremely detailed information about the nanoscale architecture and fluctuations. And because the dry mass is linearly proportional to the refractive index, you can also do mass transport studies in cellular diffusion. Now, the problem with this technology is that it requires a transmission-based geometry, and so you're limited to thin samples. And this is something that my lab has sought to overcome, the significant limitation to enable the same capabilities as QPI, but now in arbitrarily fixed samples. What we came up with was something that we borrowed from the Jerome Mertz group and tinkering with some of the math from Colin Shepard and Laura Waller. What we came up with was a quantitative oblique back illumination microscopy system, or QOBM, that comprises a very simple bright field system with epi illumination. And what we do is we acquire 4 acquisitions with these 4 illumination sources around the objective. And these images are underwhelming. They're just bright field images with a lot of out of focus. But what we can do now is, with an understanding of the angular distribution of light at the focal plane, a little bit of math that's not too complicated, we can come up with images that look like this, which is just absolutely astonishing that we can do this with a simple system. We have extremely high contrast, extremely high signal to noise ratio, and all the information is fully quantitative. This is a video going through in Z of a human brain sample from tissue discarded from neurosurgery. We can really appreciate individual neuron soma, all the processes from myelinated axons. Here we'll start seeing some cells with a little bit of lipid. We can identify lipids because they have high refractive index and they're extremely bright. And here we go, right there, that cell. And as we start going deeper and deeper into the tissue, we start seeing blood vessels and then more blood vessels. There's just a wealth of structure here. We're going about 200 microns into the tissue, which is just astonishing. Now, there are other linear technologies that provide 3D information by using scattering. You have optical coherence tomography and you have reflectance confocal microscopy. But those are fundamentally different because they acquire the backscatter field. What we're doing here is actually acquiring the forward scatter field because we actually rely on light being diffusely scattered through the tissue and then coming back at us. So that's the beauty of the oblique back illumination. So because we fundamentally acquire different frequency regions of the sample, our images look a lot more natural. So it doesn't matter
Francisco,
if
I think I would like to ask you to respect the 10 minutes and get towards the end.
Okay, I thought we had 15. Anyway, okay, so confocal reflectance, it doesn't matter if you average 16 times or however many times, you're just not going to have the same information. So I argue that really what we compare mostly to is actually nonlinear microscopy. We really have similar structural information. Yes, we're missing the colors that you have with nonlinear microscopy, but we have similar structural information. At least we're missing the colors, but I think we can improve upon that if we're smart about it. We have similar penetration depth for label-free molecular imaging with nonlinear microscopy. We're potentially faster because we have wide field illumination. And it's simple, low cost, and there's really no worry about tissue damage. So we've been using this technology and applying it all over the place. We've started looking at cells inside complex bioreactors. We can provide very specific information by looking at the fluctuations. We can look at thick organoid structures. Here, this is again rendering the fluctuations over time in color, or we can look at the time lapses. In this particular case, you have a glioblastoma organoid, about 1 millimeter in thickness, and you see these T cells basically fighting the organoids. This is just a remarkable level of detail. We've also developed fiber-based systems that provide possibilities for endoscopic and microendoscopy. We can also put this into little robots to steer them around wherever we want for like surgical procedures or to look inside bioreactors. We've investigated tissues with sickle cell disease, where we found that the refractive index of the blood vessels is different for those that have sickle cell disease versus those that don't. We can also look at the blood inside the vessels. And we've also started investigating the identification of cancer and tumor margin delineation with this technology. And we can color code the structure in red if we think it's cancer, green if it's benign, and yellow if it's something that's infiltrative tumor. And with the last minute that I have left, I really want to address this application because it's extremely exciting. This is a low-grade glioma with an infiltrative region. This is impossible to detect intraoperatively with existing clinical tools, but hopefully you can appreciate the level of detail that we can provide with the face images and that of the gold standard H&E after you take out the tissue and process it. And now we can also take our face images and translate this into H&E. And it's label-free, it's extremely fast. This can potentially be done in real time, and it's 3-dimensional. So there's a lot of capabilities here that were really beyond the reach of the imagination just a few years ago. And lastly, we now have a handheld probe that operates at video rate in real time. We've recently done an in vivo experiment in a large animal, in a swine, and this should be in human clinical trials by the beginning of this next year. So hopefully, I've been able to show you that we can also address this issue of subcellular detail in 3D. And with QOBM, we can provide unprecedented subcellular detail at video rate, volumes, very, very fast. It can be done in vivo, and it can potentially provide functional and with a little bit of AI, potentially even molecular information. And with that, I'll stop here and just leave this slide up there.
Thank you very much, Francisco. I also want to apologize. There was a miscommunication, and indeed you had 15 minutes, so my apologies for this. We will make sure we can dig deeper a little bit more with the questions and the things you presented. So I would like to ask the committee members if they have questions for our speaker.
I have, yes, thank you. So, hey, this was so cool. This was so nice. Thank you. So I have a lot of questions, but I guess I have to be respectful of other people's time. So where should I start? So from the first part of your talk, when you were talking about deep UV and its applications for sensing, you mentioned quantification of, for example, hemoglobin at the single cell level. How do you get the ground truth for that? So what constitutes the ground truth, and how do you ensure that at the single cell level your distribution is correct for the sensing?
That's a great question. So there we actually leveraged our capabilities to also do face imaging with this interferometric approach. So we compared the quantification of hemoglobin with deep UV microscopy to that of phase contrast, which has now been well established to quantify hemoglobin content. So yeah, and we actually quantify it across different spectral regions to see which one's the most accurate. And you can do a pretty good estimate with all the wavelengths below 300 nanometers, but there's actually some advantages to using 260, 250 nanometers.
Cool. If others don't have questions, so please go ahead. Yeah.
Yes, there is a question from Harry also.
Oh, yeah. Thank you for this nice presentation. So one question I had, one of the historical limitations with, linear optical microscopy in biological tissues is non-specific fluorescence. And that tends to become aggravated in the ultraviolet. And I'm wondering, you chose an example of blood cells, which are extracted from tissue. And I'm wondering if you could comment on the role that non-specific fluorescence plays in the deep UV microscopy and its application to tissues where nonspecific fluorescence would be an issue?
Yeah, that's a great question. So fluorescence was actually a big concern for us in the beginning. And actually, the large motivation for building this interferometric device was actually that this would inherently get rid of the autofluorescence because they would not interfere with the reference arm, right? And then after we said, let's make this as simple as possible by going into the system, a concern was that we would have a lot of contamination from nonspecific fluorescence. That's very, very true. And we went hunting for it. We tried really, really hard to get the autofluorescence, but we just simply do not get autofluorescence because our integration times are so short because we're doing transmission-based imaging that we just don't collect enough background fluorescence to actually be a problem. And that's actually a really important advantage here because we are much faster than autofluorescence. Autofluorescence tends to be weak relative to just the transmitted light that's going through. And so we just don't see it. It's not a contamination. It's not an issue. We tried really hard to look for it. It's just not a concern.
So yeah, I guess you showed the example of blood cells. Is this also true in tissues like organ tissues or muscle tissue and so forth?
I don't know yet. We've looked at cells, and with cells, it's not an issue. We've looked at fixed tissue sections. And it's not an issue there. We looked at frozen sections. It's not an issue there.
Okay.
If you start looking at muscles and thick tissues, then I don't know, but maybe. But
yeah.
Our integration times here is in 10 milliseconds, maybe it's sometimes 100 milliseconds, and we just don't see a whole lot of fluorescence. And it's actually microwatts of illumination that we have here. So there's just not a whole lot of autofluorescence.
And for the QOBM technique, you're using back illumination. What limitations does that pose for the sample thickness?
Yeah, that's a great question. And that's actually something that we get very frequently. It's actually very forgiving. If you have a thin sample, so with organoids, it's actually the best example because organoids can be very small. They can start out with like 100 microns in diameter, and then they can grow to be millimeters, right? When they're very, very thin, we just simply put a scattering medium on top, and then that provides the back illumination, and then that allows it to grow at whatever size it wants to, and then it transitions smoothly. So we have to provide an artificial scattering layer if it's really, really thin. And then once it grows to a certain size, typically 500 microns, that's enough for itself to produce the back illumination.
Yeah, I was thinking about the opposite limit where you have a thick tissue. Yeah.
Oh, it's not at all. So actually, let me see if I might still have a video of this thing, but I mean, we've done big brains. I can put my arm under the microscope and you can see it.
Okay.
Yeah. Yeah, it's beautiful. I'm biased, but it's great.
All right, I hear your enthusiasm. Thank you.
Thank you. I think, Aidan, you had more questions you wanted to unravel with Francisco.
Before me, Dr. Yang had some, I think, questions.
Okay,
yeah.
perfect. Dr. Yang, go
yeah,
ahead.
thanks a lot. It's a beautiful talk. I do think a label-free and low-cost microscopy will have a lot of application in medicine and biology. But Francisco, it would be great if you can elaborate on the innovation in your approach. For example, use color code phase contrast imaging. It's convenient, but it has been explored in many groups. What is an innovation contribution from your group?
Yeah, that's a great question. Of course, this is work that Professor Xing has pioneered, changing the phase images to look like H&E. I think that was really pioneering work, and even taking confocal reflectance images and making them look like H&E. I think the real advantage that we provide is the baseline technology. Here, this basically tells you the difference between the two technologies. If you have a better image to begin with, which QOBM provides compared to confocal reflectance under certain circumstances, you're bound to do better. And so our approach in translating our phase images to H&E was simply just taking the phase images and doing unpaired H&E translation. So we had to train it with a bunch of H&E. The uniqueness here is the easiness, the ease in which we can do this translation. We don't have to do an intermediate staining process in order to get that information. And our penetration depth is in the order of 100 microns with this translation, potentially even deeper if we can improve the SNR as we go a little bit deeper. And other than confocal reflectance and work by Professor Xing, there has not been any other technology that can do this in the linear regime without stains that can provide H&E images of thick tissues in 3D. Right.
Great. That's impressive. Yeah. Thanks a lot.
I think we have time for one last quick question.
While we're on this slide, if nobody else is asking, so for this 3D stack, if I see it like it's probably 50 microns to 100 microns range, right?
Yep.
The one. So how do you ensure that each slice is correct in the sense that what is the ground truth? How do you assess the quality of the virtual H&E with this 3D block?
Yeah, that's a great question. We had to fight. This was just accepted into Optic on Wednesday, so it's very recent. And we had to fight with the reviewers on that one a little bit.
I wasn't the referee.
No, I'm not saying anything. I'm not pointing fingers.
No, I wasn't.
But yeah, so we had several different steps, and actually how we validated this is very important. One, we had a panel of 5 different neuropathologists look at virtual H&E images from our animal model and from human samples, and compare that to real H&E images from the animal model and human samples. And effectively, they can identify disease at exactly the same rate. How much they like the images was rated equally. There's always a little bit of a preference for the virtual images because there's more consistent staining. We've seen this in multiple studies, actually, which is kind of strange. And then the other thing that we did is from the animal model, what we ended up doing was training a neural network to identify cancer with just the real H&E images. And then we fed it all the virtual H&E images and asked it, can you actually identify cancer? And the answer was yes, and it performed extremely well. So that tells us that we are actually recapitulating the appearance of H&E with enough accuracy and enough diagnostic quality that even a machine can get it right, and that neuropathologists can by eye visually say that it is correct. Now, in a pixel by pixel notion, we don't know what ground truth is, and that is something that is extremely challenging. I think that your pioneering work with acetic acid is really great because it has that pix-to-pix ground truth. We tried that here, but with brain tissue, there's so much deformation of the tissue that we just cannot get that acetic acid stain to be pixel matched with fresh tissue. Skin is different.
Yep. Thank you.
Thank you very much, Francisco. Thanks to the committee for the questions. So I think we can move on with the second speaker, Professor Sang-Hoon, who is at the Department of Mechanical Engineering of Washington University in St. Louis, and will present it to us about freestanding nanomembranes for photonics van der Waals integration.
Hello, can you hear me?
Yes,
yeah,
all
thanks
yours.
Okay, thanks for the nice introduction, and I'm very excited to be here as a finalist. I already so glad. And today I'd like to talk about our team's effort for the development of the nanomembrane and their application, especially based on the photonic van der Waals integration. Before starting my main topic, I'd like to discuss about my entire career. My career has been dedicated to the material innovation for optoelectronics. So during my master period, I developed the flexible electronics based on the flexible transparent electrode. Also, during my PhD, I work on the development of highlight perovskite material, especially single crystal highlight perovskite material for the high performance device, at the same time the longer lifetime. Also, after moving to the postdoc year, I work on the freestanding nanomembrane and especially solar cell application for space vision. And after moving to the Washington University, I work on I keep developing the freestanding nanomembrane. Also, I have been working on discovering the new types of physical coupling by designing the new hair structure. Also, we developed the new types of application based on the vertical integration. So that's the kind of summary for my entire career. And second, let's move to the motivation part. The tour to the future application, 3 transformations have been required. First thing is about the from being rigid to the being flexible, and second thing is the single functionality to the multifunctionality. Last one is the layer integration to the vertical integration. So people look for the methodology to reach out to realize these three all together at the same time. And we can find the answer how we achieve those three from this fundamental graph. As you can see, that when we reduce the thickness of device and material, then it eventually leads to the extremely small stiffness, so that it can be the flexible and conformal as well. Also, this stiffness concept is very closely related to the required adhesion energy, which means that we are easy to making vertical stacking using very thin nanomaterial. So this one is the device that I made in 2014. At 15, as you can see, that when we make the very thin device, which is the 1.5 micrometer, then we are even make a very good conformal contact on uneven substrate. So we can say the thinner is to realize the flexible conformal device, also to realize the multifunctional device by stacking the multiple material. Also, we can integrate the vertical manner with the multiple functional device as well. So our team have been working on developing two different regions for that ultra thin material. First one is the conventional material, which is based on the architecture goals. Second section is about the large scale high quality 2D material. So let me start with the first part first. We need to take a look why it was so difficult to make the very thin freestanding 3D membrane. That mainly because the problem comes from this strong covalent bonding at the interface. Because of this strong covalent bonding, we are not easily selectively separate this ground material from the substrate. And substrate has a usually 1,000 times thicker than ground material. So what people try first is that this covalent bonding at the interface problem. So people try to grow this single pixel material on van der Waals terminal surface. But in that case, another problem happened. In case of the covalent terminal surface, this side orbital is not really fully saturated. So thermodynamically it's unstable stages. So they want to make a bonding with an additional atom. So it means that the supersaturation degree on this side is pretty high. So critical Gibbs free energy is extremely low. We are able to obtain the periodic potential for the nucleation. On the other hand, on the van der Waals terminal is exactly opposite case. So we are not able to, this part is already, orbital are already fully saturated. So the supersaturation degree is extremely low and the critical Gibbs free is high. So periodic nucleation easily form, it's not easily formed on the surface because its thermodynamically already stable. So to make it happen, growing single pixel material on this van der Waals terminal surface, it may be we need to increase this critical Gibbs free energy again. So what we try is that this van der Waals, people usually try the very thick van der Waals layer to grow the single crystal material, but what we found out is that the potential fluctuation can penetrate through this this van der Waals layer, so that we reduce the thickness of this layer material, and we try to grow the single crystal material on that van der Waals terminal surface. And it turned out to be very successful. As you can see, that we successfully grow the single crystalline gallium arsenide material on van der Waals terminal surface. And as we targeted earlier, now we no longer has a covalent bonding at the interface, so we are able to selectively separate this ground material so that we obtain the freestanding nanomembrane. Actually, at the beginning we thought that it's universal approach, so we are so excited. In thin film, optoelectronic thin film region is kind of dream to realize a very thin nanomembrane with the multiple different material. But previous methodology have been introduced is quite limited because of some fundamental reason. So what we thought is it may be the universal approach. So we are so excited. So we try to apply this to the other material system. But when we tried to germanium, then it was not really successful. As you can see, when we measure the diffraction pattern, it turns out to be multicolor. It's the EPSC, the color mapping. Monocolor is the single crystalline, and the multicolor is the polycrystalline. Whenever it involved with the germanium, they all produce the polycrystalline. So what we kind of disappointed to see that result, but while understanding the situation, we found out another interesting phenomena. We did a DFT calculation, and what we found out is that the compound semiconductor can produce this kind of very strong potential fluctuation even through the monolayer, the band-arrest material, while this elemental semiconductor cannot do that. So these two different material has a different polarity property. So what we conclude is that the polarity matters for that potential fluctuation through that band-arrest material. So another thing, to expand the material library, we need to grow the higher material groups. In case of higher material groups, we need to carry the strain energy, unlike the homo material case. So, but eventually this strain energy, when strain energy go beyond that limitation that material can hold, eventually it start producing the dislocation. So handling that dislocation is quite important to have a high quality, high performance device. But in our case, it doesn't have what we expected. It wouldn't follow this situation. That mainly because the dislocation, original dislocation comes from this the strain energy, and strain energy comes from the covalent bonding. But in our case, it doesn't have a covalent bonding, so that we expected it wouldn't follow that scenario. And what we found out is that there is another scenario exists, which we call the interface sliding situation. And interface sliding situation has a much lower threshold energy compared to the threshold energy for the dislocation introduction. So that eventually we reduce the 8 times smaller defect density on our system. By applying this kind of approach, we currently, so far we have been working on many different material, from optoelectric material to the magneto-optic material and electro-optic material too. And even we succeeded in the scaling up to the 3-inch, and this one is a 3-inch, and recently we also succeeded in 6-inch demonstration based on this principle. And second part is the 2D material, large scale 2D material case. In case of 2D material, is kinetic control of 2D material is much more difficult compared to 3D material. 3D material case, it has a room to experience this all kinetic process, but 2D material case, it doesn't allow this kind of process. Whenever we have additional atom, that it turns out to be irregular 2D material. So property could be not uniform. So what we thought is that having monolayer by growth is extremely challenging. So we try to grow the if we find a way to make this multi-layer into monolayer, that could be the good solution. So we designed the mechanics to produce the monolayer from multi-layer, so that eventually we produce the large scale, wafer scale monolayer 2D material. And we measure the fluorescence sensor. Monolayer has a direct band gap compared, while multi-layer has an indirect band gap. So we observe the substantial fluorescence boost from this split 2D material. And we succeeded in demonstrating all other 2D material using this splitting technique. And while we are making, based on this 2D material, we experience eventually we need to find a good way to grow the high quality 2D material. Because even if we control the thickness, still the quality of 2D material determined by the growth. So we look at what kind of problem exists in this kind of 2D material community, and what we found is these 3 challenges exist in 2D material: single crystallinity and large scale, and layer by layer controllability at large scale with a single crystalline, and higher structural formation at large scale. So we really need to find a way to achieve these 3 at all together. So we took a look at the most fundamental problem again, and as I explained a little bit earlier, the biggest problem comes from the difficulty in controlling kinetic of 2D material. Because the random nuclei easily happen, and it always requires extremely long growth time. So what we thought is that many of applications doesn't really require the entire wafer. Instead, they only need the periodic high quality material. So we designed a structure made of two different materials. Those two different material has a different absorption rate. One material has a high absorption rate for the target because another material has a low absorption rate for the target material. So that all the kinetics happen on the target region, so that we can define the seeding area also. Now we don't really need to grow the entire area, so we can. It doesn't require the extremely long growth time, so we can shorten the growth time. Also, there is a step between these two regions, so it can prevent formation of lateral hair structure. And we did a DFT calculation, and we found out that the silicon dioxide and aluminum dioxide, they have a good combination to confine that structure. Also, silicon dioxide and hafnium dioxide, they have also good nuclear selectivity for this purpose. As you can see, we successfully confined the seeding area, but when we have a larger pocket region, then it cannot secure the single seeding within this target region. But when we further reduce, we thought that this one is still required a longer growth time. So we further reduced the dimension of the confined region, so that eventually we can secure the single seeding inside of this target region, and eventually you can produce a single crystal material at a large scale. Also, because I skipped many of the details, but we succeeded the layer by layer control. Also, we successfully grow the vertical hair structure. And based on this one, we kind of had a new types of material building blocks. So we work on the designing new types of hair structure that didn't exist before. Because the structure is completely new, so we have a lot of chance to explore the new types of new types of physical coupling as well. So we can currently explore the many of different physical coupling in this region. Especially, I got trained for the optoelectronics region, and currently we expand to the pure optics region as well. So we recently published the one review paper regarding this topic. Now, actually, people already know which kind of structure are ideal for the photonic device, but there was no way to realize that photonic structure. Now we have a much high freedom to design that structure using our technique. So we can design the ideal structure, and we can see the high performance based on that one. Because of time limit, I didn't include that detail here, but we recently observed the extremely high electro-optical modulation based on this structure. It's especially 20 times better than the state-of-the-art region. Also, we recently successfully demonstrated vertically stacked LED, the micro LED device. Usually, conventionally, they design this RGB in the lateral manner, so that there is a limitation to reduce the density of this one. So we even achieved the highest RGB pixel density using this vertically stacked micro LED. Also, we designed the new types of the device application based on this photonic van der Waals integration, such as this one is an in-sensor computing made of 3-5-based optoelectronics and silicon-based computing, so that we can receive the information, optical information, directly through this 3-5 material-based photonic sensor. Right after receiving that one, we can compute that result using this silicon-based computer. Also, we did a similar thing using 2D material, because still 3D material has a kind of limitation. So we further reduced the thickness by using the 2D material, and even we achieved to realize the total 6-layer functional device based on this 2D material too. So that's kind of pretty much. So my team worked on this freestanding nanomembrane to help kind of that transformation for future optoelectronics. Before closing, I really like to thanks to all of my people, my students and postdoc. They're very self-motivated. I really thanks to them, and also I'd like to thanks to all of my collaborators and sponsors. Yeah. That's it for my presentation. Thanks.
Thank you very much for this nice presentation. The floor is now open for questions from the committee. Would anyone want to start? Yes, Minchu, please go ahead.
Yes, probably I can start. Beautiful work, very interesting results. For your 2D nanomembranes, you first grow several layers of 2D materials, and then you peel off. Yeah, I think, yeah. So how you control the layers you are peeling off? I mean, how you can know that finally you only have one layer or two layer, or how many layers, and how precisely you can control them?
Yeah, that's very good question. Because of time limit, I skipped the detail. So actually, we designed the structure by having the binding energy contrast. So for example, we grow the multi-layer here on substrate, then we deposit the metal here. Then it has a 3 different interface. One is the metal and 2D material, another 2D material and 2D material. The last one is 2D material substrate. In this case, substrate and 2D material, that interface has a weakest interface. So when we try to apply the energy here, then separation happens at this very bottom side. And afterward, we deposit another metal at the backside. Then these two interfaces has a very strong adhesion energy. So when crack propagate, then it just naturally go over this monolayer because of these two interfaces has a very strong interface toughness. So it's kind of self-limiting process, and eventually we can produce the monolayer by using this technique.
Okay, thank you.
Yeah. I see Harry has a question.
Yeah, thank you for this very nice talk. Your talk focused primarily on the fundamentals of the exfoliation process and the interaction between 2D materials and 3D materials. And in photonics, I would say the usefulness of this would be also related to the performance of devices and device applications. I wonder if you could say a little bit about that, about about the impact, both broadly and also related to the materials that you described in your talk here. This is a body of work that I know that you began also as a graduate student in the lab of Professor Ji-Won Kim, and maybe you can also distinguish a little bit some of the things that were done here in your lab from the work that was done in Professor Kim's lab.
Yeah, that's very good point. Especially making material side is all done during my training year from the Professor Yang lab at UCLA and Ji-Won Kim's lab at MIT. And that's but after moving to WashU, I more focused on the physical coupling coming from this we call the artificial header structure. This as you can see, this is independent work, and also we designed the new types of header structure made of that freestanding nanomembrane. I didn't include the detail here, but we recently designed this photonic structure made of the freestanding barium titanate on the silicon the waveguide. So people usually work on the lithium niobate recently because the lithium niobate has the high focal coefficient. But what we observed is that the barium titanate even has the 50 times stronger the focal coefficient, so that we can further make the more energy efficient device out of that one. And fundamentally, it has a 50% higher performance. But what we observe is the 20 times high performance device. And not only this on the because it is only 50-minute talk, so I couldn't include the detail. And we observed many of different property. For example, we also observe the dielectric relaxation can be modulated in this system. It's completely independent work, and we observe that it can enhance the capacitance property, so that we can include more charge, so that we can control the material, the density of state, so that when we try to incorporate it photonic device, then also we observe the more energy efficient device. Okay.
We have time for one more question. Yes, Dragomir.
Thank you very much for your wonderful talk. So it's a really exciting technique and what you can do with it. So just to follow up on the device side, do you know if anything with phonon propagation on these films, on these nanomembranes, has been investigated either by your group or by your colleagues, and what has been the state of the art there?
Oh yeah, that's also very important things we are investigating recently. That we actually we have an interest in that. We are preparing another paper based on that what you mentioned. That we what we found out is that we can enhance the piezoelectricity based on this freestanding nanomembrane, especially when we reduce the thickness of material. Then it has a more freedom to propagate the thermal energy out of that free space environment, so that we can even further enhance the piezoelectricity by just reducing the material thickness. And also we can treat the surface with 2D material with 3D freestanding nanomembrane, so that we can control the heat capacity within that region. So we even further enhance the piezoelectricity. So far, we need to further investigate what's the main reason we can achieve such a high piezoelectricity. But we can control the phonon property using this material.
Thank you.
Thank you very much, Sang-Hoon. And we will now move on with our next speaker. The next speaker will be Professor Junjie from Tsinghua University, and he will be talking to us about implantable optoelectronic devices for neural modulation and sensing. The floor is yours.
Hello, can you hear me and see the slides?
We can hear you. I think the slides are coming. Yes, now we can see the slides. Perfect.
Okay, great. Okay, great. Let me see. Okay, I think I just go ahead. Hello, everyone. I'm Xing from Tsinghua University. It's my great pleasure and my great honor to present our research here. In the past few years, my group mainly focused on the development of implantable optoelectronic devices for the brain research specifically, and how to use these devices to modulate and sense neuron signals in the brain. And here's the brief background. And nowadays, the brain science study has become an emerging field and attract worldwide attention. And the advances for both neurological disease treatment and the novel neuromorphic computing systems requires the new technology to understand our brain. And here I would like to quote from the Nobel laureate pioneer in the neuroscience, Sidney Brenner, and he once said, the progress in science depends on new technologies, the new discoveries, and new ideas, probably in that order. So therefore, the new techniques is always considered to be the driving force, the key driving force in the scientific discovery. And in our group specifically, we focus on the development of advanced electrical and optical devices that can be implanted into the brain system. And on one hand, we want to use this device to generate electrical and optical signals to interact with the neural system, to monitor and control neural activities for some fundamental neuroscience studies. And on the other hand, we work closely with the clinical experts and hope to use this device for disease diagnosis and therapy in the future. And I am from the electrical engineering department, and with a background of study optoelectronic materials and devices. And we know that the conventional high performance devices are mostly based on the bulk and the rigid substrates. And the key challenge here is how to integrate this device with the soft and flexible biological system, especially the brain tissue. And we use design and fabrication strategies to realize the thin film microscale materials and devices. And this device, they can have a form of flexible membranes that can be attached on the soft tissue, or a needle shape that can be embedded in the neural system. And we can implant these microscale devices into the brain. On one hand, we use this device to generate optical and electrical signals to interact with the neural cells and manipulate the neural activities so that we can better control the nervous system. Here are some recent works. We developed devices for optogenetic and non-genetic stimulations in the brain system. And on the other hand, we can also utilize these implantable devices as sensors to detect the electrical, optical, or chemical signals inside the brain so that we can better understand the neural systems. And there are also some recent works. We developed the implantable sensors to record, for example, the calcium fluorescence, temperature, or dopamine signals in the brain system. Okay, in today's presentation, I would like to share with you a recent, actually unpublished work that, as an example, to showcase how we create the implantable devices to detect the brain activities. And specifically here, we develop a photometric probe that monitor the oxygen in the deep brain. This oxygen sensing mechanism is based on the fluorescence change of a phosphor coating on the implantable probe. And we designed a circuit to operate this device wirelessly in freely moving animals. And as a background, the oxygen probably is the simplest molecule we can find in our body, but it's crucial for the brain function. And our brain only comprises only about 1 to 2% of the body weight, but consumes more than 20% of the overall oxygen and the energy. Therefore, our brain is the organ most sensitive to hypoxia, which means a low oxygen level. And many diseases, for example, including the brain injury, the stroke, seizure, and brain tumor, will affect the oxygen level in the local brain region. Therefore, the technology for real-time oxygen monitoring is very, very important. So there are some existing technologies for oxygen detection in the body. For example, the measurement of the blood oxygen saturation. We are very familiar with this kind of finger clip pulse oximeter. This detection actually is based on the optical absorption difference between the oxygenated and deoxygenated hemoglobin, the HB and the HbO2 in the red and the near infrared regime. Actually, this is an indirect measurement because it's only detected the hemoglobin, not the oxygen molecule. And another commonly used approach is based on the functional MRI, and which can also image the blood flow in the brain. It is based on the different magnetic properties between the HB and the HbO2. And so therefore, this is also an indirect measurement of the oxygen. Okay, and there are some ways to directly measure the oxygen in the brain. For example, this technology is based on the polarography. This is an electrochemical technique. They use a Clark-type electrode inserted into the tissue. This is actually a very bulky electrode. It includes a lot of components: the diffusion membrane, electrolyte, and the working and the reference electrode. So the response of this device is also relatively slow because it will have to allow the oxygen to diffuse into the electrolyte. And in addition, this device actually consumes oxygen in the tissue when it operates. Also, this is a wired system, and it's connected with an instrument for the data recording through a wire. And this cannot be used for the freely moving behaviors. And to summarize, we need a more compact wireless method for the oxygen detection in real time. So here I present our approach. And in this work, we developed the wireless photometric probe for oxygen monitoring in the deep brain. And the probe integrates the thin-film microscale optoelectronic device, including the LED and detectors, and the phosphor coatings for the oxygen sensing. And here are some more details about the microprobe system. Okay, the key component here is the oxygen sensing film based on the PT-TFPP in PDMS. This PT-TFPP is a platinum-based complex, and this molecule has an absorption peak in the violet region around 400 nanometers, and has an emission in the red region around 650 nanometers. And also this PT-TFPP has phosphorescent quenching in oxygen, which means that this red emission intensity is actually very strong in a low oxygen environment, and we can use that for the oxygen low oxygen level sensing. And to excite this phosphor, we use a violet LED as a light source. This LED structure is a indium gallium nitride diode grow on sulfide substrate, and we use the epitaxial liftoff and transfer printing technology we developed to release this thin film LED from the original rigid sulfide substrates for heterogeneous integration. And to detect the fluorescent signal, and we use a photodiode. And similarly, this detector is a indium gallium phosphide diode grow on gallium arsenide substrates. We similarly use a epitaxial liftoff and the transfer printing method to release the thin film detector from the original gallium arsenide. And both the LED and detector here, I show here, they have a geometry about 100 to 200 micrometers, and the thickness only a few micrometers after the liftoff. And the violet LED have a emission around 400 nanometers, and the detector is optimized with a thin film filter coating and shows selective response in the red region, so that we can see the device performance has matches very well with the optical absorption and the emission properties of this PP molecule. And finally, and all these components can be heterogeneously integrated on a thin film flexible substrates, and the probe structure is formed by laser milling. Because of the PP coating on the probe surface, we can see that this shows a very bright red emission color when the oxygen level is reduced. And the circuit is designed to remotely control the probe and wirelessly transmit the detection signal. The circuit is powered by a rechargeable battery and controlled by a Bluetooth chip. And the circuit has a size about 1 to 2 centimeters and weight less than 2 grams. It can be mounted on the head of a freely moving mouse for the real-time oxygen monitoring. And first, we tested the probe performance in an ambient environment. We flowed different mixed oxygen and nitrogen into a gas chamber, and we can see the probe records a strong signal increase at low oxygen pressure. This indicates that this probe is very sensitive to the low oxygen level. And the inverse of the fluorescent intensity is proportional to the oxygen pressure. This follows the standard classical Stern-Warmer relationship. And here shows the dynamic response. We alternate the oxygen and the nitrogen flows in the chamber, and we can observe the probe have a dynamic response to the gas environment, and the response time is less than 1 second. And next, we move to the in vivo experiments, and this probe is implanted into a mice brain, and we provide the animal with different inhaled gas with different ratios of mixed oxygen and the nitrogen. And we can see that this is the gas flow, and the probe can dynamically monitor the oxygen pressure change inside the brain. And we can compare the performance of our probe with a commercial fiber-based wild sensor called the NeuroFox, and we can in the same brain region, and we can see these two systems have a very similar dynamic response. And another advantage of this wirelessly operated probe is that it can allow the oxygen detection among multiple animals behaving in the same environment, so that we can use it to study more complex behaviors and the activities. And to further explore the potential of this probe and study some real neuroscience problems. And here we use this device to detect the oxygen activity in a brain disease model. And in this model, we insert the electrode into it's called the hippocampus region of the mice, and apply a electrical stimulation to induce the seizure activities. And we use our oxygen probe to monitor the oxygen pressure change during the seizure event. And here shows the results. And each kindling here means a short pulse electrical pulse applied in the hippocampus. And associated with this each kindling, and there is a drop of oxygen level in the brain tissue after about half or 1 minute after the stimulation. And here is a short movie. Let's see if I can play the movie here. And yeah, in this experiment, we simultaneously recorded the electrical activities of the neurons called local field potential, and we apply let me replay it. Okay, we apply a short pulse here, just a 1 to 2 second pulse, and after the electrical stimulation, we can see the intensified strong LFP signals, and the mouse shows strong seizure events. And following the LFP increase, there's a significant drop of the oxygen level in the brain tissue associated with the cell consumption during the neural activity. And we can see that there is an interesting time delay among the kindling and the LFP and the oxygen level change. So that our probe can help us to try to understand the coupling between these neural activities and the oxygen consumption in the local brain tissue. And we can also later point here. We can also implant this probe into different brain regions and investigate how the oxygen pressure change in different locations of the brain under the same stimulation pattern. And as example, we can see that we implanted the probe into a CS3 in the hippocampus, and also the M1 is in the motor cortex. And we can see that these two regions show a different temporal response and oxygen dynamics associated with the same kindling events. And here we compare the results of the oxygen pressure change and the blood oxygenation. As we mentioned, there are different signals. In this experiment, we use a fiber optical system to measure the optical absorption change in the red and the near infrared region in the hippocampus, so that we can determine the ratio of HB and HbO2. And we can see that when we supply the animal with different gases, the blood oxygen response is similar to the oxygen pressure we presented previously. However, when we apply the electrical stimulation and in the hippocampus to induce the seizure event, we can see that this blood oxygen response is actually increased. This is due to the neurovascular coupling mechanism, and this is totally different from our recorded oxygen pressure data. Okay, and we can also look at the fMRI results in the literature, because we are not doing the now experiment, but we check the literature data. We can see that previous works also investigated the blood flow associated with seizure event. We can see that also they observe an increased blood flow and oxygenation during the seizure. This is also different from our recorded tissue oxygen, which we decreased. Therefore, this tissue oxygen and the blood oxygen are two different signals, right? While the ratio of the HB and the HbO2 indicates the supply from the blood vessel, and the oxygen pressure in the tissue in our detection actually is more closely related to the local consumption. These two signals actually have different responses. They are actually complementary to each other, right? By studying both signals together, we can have a better understanding of the coupling between the brain activities and the oxygen supply and the oxygen consumption. Okay, and in the system level, we can further optimize the wireless circuit module. The first version is based on a battery powered system, and which limits the circuit size and the weight. And in the improved version, and we can replace the battery with a thin film inductive coil, so that the full circuit can be completely embedded underneath the scalp of the animals. As shown here, the system can be remotely powered by an inductive coupling with a external antenna transmitter, and the battery-free circuit can enable long-term monitoring of oxygen levers.
Okay, I would like to yes.
Sorry, yeah, I have to conclude. Okay, here I present a optical photometric probe based on this optoelectronic devices for oxygen sensing in the deep brain, and help you to use this work as an example to show you our general interest, which is how to use the device, implantable devices and the materials to help us to modulate and sense the neural activities and understand the brain system. Okay, and finally, I want to thank our research group and our collaborators and our fundings. And finally, thank you for your attention, and looking forward to your feedbacks.
Thank you very much for this nice talk. So the floor is open now for questions from the committee. Aidan.
So thank you for this beautiful presentation. Can you hear me?
Yes, sure, yeah.
All right. So at the very beginning of your presentation, you motivated us on some of the human use applications. So regarding some of those applications that relate to humans use, can you tell us a little bit more about biosafety, biotoxicity, and some of the fouling issues regarding especially the phosphorescent probe that you have there? Is it safe?
Right.
You know, is it going to be safe for humans? And what are some of the challenges that you see around that?
Yeah, sure. Moving forward in the future, if we want to apply this kind of technology for clinical studies, this biocompatibility issue and biosafety issue will be critical, right? And we can see this is an implantable probe, so that it will generate some damage in the tissue, of course. And we carefully apply some biocompatible coatings on the probe surface, which is transparent and also biocompatible, so that it will reduce the immune reactivity. So that we did a systematic study of the immune response of the tissue surrounding the probe. And I do not have I didn't have time to show all the details here. And we try to compare it with some conventional probes, for example, those commercial implants and optical fiber-based implants, and they show a similar reaction response. And moving forward, another approach we can explore is further reduce the geometry of the device, so that we can minimize the damage. And another
more than the physical damage, I'm curious about the toxicity of the phosphorescent probe. Is it a safe material for human use?
Yeah, we actually I think that this phosphor molecule haven't been approved for clinical study yet. But in our case, we embed this phosphorus molecule in the PDMS matrix, and we test the stability of this phosphorus in the aqueous environment, and we just immersed in the water for a long time, for 1 to 2 months, and we didn't find any observable leakage. So the molecule actually is stable inside the encapsulating coating. It will not cause any leakage. And of course, for better for clinical applications in the future, which we will have to do more serious investigations to understand this biosafety, of course. Yeah.
Thank you.
I think there was a question from Lan.
Yeah, thanks for the beautiful talk. So in your work, you use a photometric probe for oxygen monitoring, but have you considered some other factors such as temperature that might affect fluorescence and affect your measurement?
Yes, this fluorescence molecule is sensitive to oxygen, but it will also be maybe affected by, for example, the environment temperature and the pH values and a lot of factors. And we tested the temperature stability of this phosphorus, and we find that it has a slight change when we change the temperature, but it's relatively stable in the body environment between, for example, about 35 degrees C to 40 degrees C. We tested it's actually quite stable in this small temperature range. And in our body environment, so we know that temperature will not change too much. Usually just below about 1 to 2 degrees C. Yeah.
Okay, thank you.
More questions from the committee?
Maybe I can ask one question. Thank you. Very interesting work. Hello. I wanted to ask you if you could distinguish your contributions into this field. It's a very exciting application of electronics and optoelectronics, using also light to monitor. But it would be interesting to hear actually what is your main contribution and what your group has demonstrated in a groundbreaking way.
Oh yeah. So the one thing we are trying to emphasize and distinguish here is that we are actually measuring the oxygen pressure, partial pressure of oxygen, so that the conventional techniques mostly rely on this non-invasive method to measure the blood oxygenation, and we actually show that these two signals actually are quite different in some events. For example, in the disease seizure model, they actually have quite different responses, and it's actually important to monitor both so that we can understand their difference, because some abnormalities can only be observed by measuring the oxygen pressure instead of doing the blood oxygenation, right? And another unique property, performance of our probe compared to the conventional probe that I didn't show here. We have a list of table to compare our technology with the existing approaches. For example, our probe have a faster response compared to those conventional electrochemical probe because they rely on the oxygen diffusion, so it's actually relatively slow. And also the geometry of our probe is actually smaller compared to the conventional probe. And more importantly, our system can be wirelessly operated, right? So it can be easily mounted on these small animals for the field behavior studies.
Thank you.
Thank you very much. So I think we can move on with our next speaker.
Okay,
who will be Arka Majumdar from the University of Washington, and he will presenting to us about software-defined meta-optics. So Arka, the floor is yours.
Great. You can see my screen and hear me?
Yes, all good.
Okay, good. Yeah, thank you. And it was great to be here. My name is Arka Majumdar. Most of the work that I'm going to talk about, which we term as software-defined meta-optics, you can find from my website. This is my email ID. First, I'd like to acknowledge my team and different funding sources, both federal funding as well as industry funding, and several collaborators, particularly related to this project. So before I start, let me tell you very briefly, my group is working on several different things, and we have a broad research portfolio. Fundamentally, we are interested in the light matter interaction. We work with nanophotonics. We pattern, design, and fabricate optical surfaces with unprecedented properties that includes maybe manipulating wavefront for optical information processing, getting stronger light matter interaction with different material like 2D material, phase change material, solution process materials, a few different materials that we are looking into. But I am not interested purely on the curiosity-driven fundamental research. I am always drawn by the applications, and in my group, we are trying to figure out with this light matter interaction, what are the applications we can enable? So one of the applications that really I'm pushing for a long time, that can we actually interact strong photon-photon interaction? This is the problem that I actually showed in my PhD, that this can work fundamentally. There's no fundamental limitations, but how do you scale it up? And it's one of those problems that almost everyone kind of gave up, but I'm still trying. I'm also working on this hybrid integrated photonics. So silicon photonics is really coming of age. But can we put new material, specifically phase change material with silicon photonics to get next generation communication system? And this is actually some of the work that we are doing with Intel. We are putting actual PCM in Intel silicon. We're also looking into kind of optical implementation of some of the machine learning framework in more like an optical front end and the computational back end. And this optical front end will be free space. We're also thinking about how can I actually bring nonlinearity there using maybe image intensifier, like solid state image intensifier, as well as like saturated absorption coming from atoms. And then this one, which I'm primarily going to focus today, is this nanophotonic computational image sensor. An idea is this nanophotonic technology, can we use that for compact image sensors? And your performance will be lower. Can we get back those performance using computational backend? So that's really kind of what we are interested in. So if you look into this problem space, the compact camera, there are, other than camera, there is a significant progress in the field of computational imaging and computer vision. And these are falls primarily under computer science community. So let me tell you like how it essentially kind of, you can think about that in a pipeline. So computational imaging, you use a low-end optics, maybe a single surface or a random material surface to get something that doesn't look like the actual image. And then you turn on computation to get back the image. Goal is to create a very, very small compact sensor, cheap sensor, and you're essentially transferring the work of optics into computation. For computer vision, we are looking into a slightly different problem. We want to capture a very high fidelity scene. That's why I call it high-end optics, so that I can turn on my computation, feature extraction, pattern recognition to make a decision. And if you look into these two problems, they're primarily happening in the computer science domain. The optics are relatively untouched. And that essentially what boils down to that often you have a huge amount of latency, huge amount of power consumption in the computational block, which essentially precludes real-time operation. So what we are interested for quite some time is, can you reduce computational complexity using free space optics? And one of the examples you can think about, you start from the left-hand side, there are 4 scenes, you want to go to the right-hand side, that you want to capture a nice image, you want to get depth, maybe you want to understand the cars, and maybe you want to understand the whole scene, right? And the goal is to use an optics to get into an intermediate stage, as I am showing here, such that when you turn on your computational block, it takes less amount of power. So you're not doing all optical. We are doing optics, we are doing computation, but the computation can help optics. Now, of course, how do you get this optics? We need to design new kind of optics, and ultimately, can we co-optimize the optics and computation algorithm for solving? These are not just only 4 problems, that all the sensing problems you can think of where you're using an optics and computation, can we do them together? That's the really kind of the basic idea we are going after. And these kind of ideas are not necessarily new. People have proposed this even back in 1989. David Stark also talked about it. The thing that maybe people have not looked into also, that can we simplify optics using computing? Like, we are still designing the optics. It's not like we're taking a random surface. So in some way, optics helping computing, computing helping optics. Together, can we reduce the complexity, reduce the latency, reduce the power, reduce the size? That's really the basic premise of this problem. And to solve that, we are looking into meta optics, which is essentially kind of a sub-wavelength diffractive optics, which most of you already are very well aware of. And really the 2 benefits that I think meta optics gives us from a very purely engineering point of view, we can make them with single stage lithography, because we can get different phase shift by just patterning things by patterning the same thickness by changing the lateral dimension, and we don't have any higher order diffraction because of the sub-wavelength periodicity. Okay, so I told you this whole synergy of computation and meta optics are important, and you can do many different things. But my group, or the only work I'm going to present, it's like I will say maybe a very small portion of my work, but I really want to focus on this, that can we capture a good quality image using meta optics? And the reason I'm kind of really focusing on that is because I feel that it's easy to do something different. You can do like a lot of different things, but can we actually do something that is a very, very well-known hard problem, very well-known difficult problem? Can we solve it? That's kind of really why I chose this topic for today's presentation. So meta optics has very strong chromatic aberration. All of you know that. It's nothing to do with meta optics, it's the diffractive optics. We have very strong axial chromatic aberration. If you want to take an image, you're going to get something that looks like kind of really chromatic. And people have tried to do some dispersion engineering, but there is some fundamental limitations. I like to point out that Federico, who is a pioneer in the meta optics, in 2022, in Nature Photonics interview, basically said this is the biggest limitation, that it cannot be done. We cannot get large aperture and achromatic operation. And I'm going to show you today that it actually can be done. Okay? So let me start with something that we first did, is extended depth of focus, to kind of get you the idea that what is the basic fundamental principle behind it, and then we'll go to show you the final results. So the idea here was that meta lens focuses light at different points. So if you, let's say, put a sensor here, green gets well focused, but red and blue are not. But can we actually create an optics where instead of putting light in one point, you put light in a line, so that red, green, and blue all come in the same way? They're all blurry, but then you turn on your post-processing software to get back the final reconstructed image. So to do that, we went with some very simple idea of adding a cubic phase mask, which people have looked into back in like 1995. And when you add a cubic phase mask with your ordinary lens, you get an extended depth of focus. So you can find that your focal spot is not in one point. You can get a long elongated line. And what it does, like if you look into, let's say, meta lens, like hyperboloid meta lens, when there is no cubic term, green is well focused, red and blue are blurry. Whereas when you add this cubic term, you get something that doesn't look like a focus at all, but they look similar. They look all similar. The second point that's important to recognize that when you use this kind of meta lens and you create the modulation transfer function, you get a lot of zeros. And when you have zeros, that information is never captured. And if the information is not captured, you computationally cannot correct it. So we showed, like with our approach, we can actually have a much larger number of special frequency being captured. We do take a hit in the MTF of the zero frequency, but these are fairly well above the noise levels we can get back with computing. And this is what we did back in 2018. Now, this was our result. This is the ground truth. You can see with our case, we can get a somewhat blue sky, some stems. If you just use the singlet like chromatic meta lens, this is how it's going to look like. So definitely we solved the chromatic aberration, but the image quality is really not there. And this has been a problem. If you look into the imaging that people have done back in 2018, these are all solved, like some chromatic aberrations are solved, but the imaging quality was not there. So to solve that, we started doing this end-to-end approach that co-optimized the optics and computational backend. And this is our current result. This is an unpublished result, but we have this 1 centimeter meta-optics. This is the first time we can actually compare a refractive to meta-optics, like real apples to apples comparison. I can get a 1 centimeter refractive with the same F number, and we compare them. We can put both of them directly on the camera. And this is the displayed result. Like, for example, like in an OLED, this is how our refractive looks like, and this is our end-to-end looks like. We also have an EDOF version. But I'd like to point out here, let's look at this side, right? In the refractive, at a large field angle, we get a blurrier image. We actually get a much better image. This is the first time a meta optics is beating refractive, at least in one aspect when the full color imaging is concerned, okay? So why does it work? And this is something that we are trying to answer for quite some time. And it turns out that the optics that we have, like if you think about refractive, you have a fairly nice point state function. This is before you capture, so no computation is done. But as you go above larger and larger angle, you start seeing a blurrier, bigger spot as we expect from spherical aberration, right? But our optimization essentially is trying to uniformly make the PSF as small as possible over all the wavelength and all the angle. And this is exactly what we have. If you look into these points are slightly bigger than these points. But overall, we get a much narrower, like for example here, a much narrower size than what we can get refractive. So this really the design insight we came up with. But then as we are making this thing, we have to make sure that we have a large tail so it doesn't go to zero, because when we get bigger PSF, then it goes to zero. So we have the PSF that may be a different shape, optimized over all the things to make sure that we have enough information preserved. So when you do computing, and this is the kind of result after computing, we're comparing MTF, and this is a little bit, I don't have time to really go over this, but like you should think about like with the error bar. So these are the measure, and after computing, we develop a new method to actually do that. So we can get like actually 30 lines per 30% contrast as 125 lines per millimeter, which is very close to actually the commercial cameras that you can get in the cell phone or laptops. We can get that kind of image quality using a single meta optics and computational backend. And the computational backend is a very simple winner deconvolution. Now, if you do neural network-based computational backend, you can get even significantly better image, as you can see here. This is taken with an iPhone camera. This is taken with a neural network backend. The only issue is the neural network backend takes a significant amount of time. So that's something that we are working on. But with the winner deconvolution, this is one example. You can actually create an image or video in real time. There is some blur, there's no question about it, but you can see that we can probably identify the people that if you know, you can see the people, you can see the grass, you can see the buildings, you can preserve the color quality. So that really brings us to my last part of the topic. So we are realizing that we want to understand the design rule, and the design rule is do MTF engineering. You make the MTF, trying to make a focal spot for all the color. We want to maximize the volume under the MTF curve over all wavelengths and all angle, making sure they are similar. We have a code that's publicly available now, and you use that to do actually broadband thermal imaging. So this is the complex scatterer and simple scatterer. Our pipeline can actually handle both kind of scatterers, and these are some of the imaging in the wild. So completely ambient light. This is the refractive, and this is our MTF engineered approach. The refractive is really ground truth. This is like really thick. So if you compare the thickness, this is 100 times thicker than what we have here. And this is the hyperbolic meta lens. So we are actually getting significantly better image quality than what we can get from hyperboloid. But I like to point out here, this is one of the limitations of computational imaging. If you have too much noise, then it doesn't work very well. And in the thermal, this LWIR, we are actually using an uncooled boson flare camera, which has a higher noise than the visible image. Now, I told you primarily on this full color imaging, but we have actually done very focal imaging with static optics and computing. We have done spectroscopy, depth sensing, also like we are looking into some of this optical neural network or meta-optical encoder. So just wanted to kind of show the progress that we made on this particular problem. As I said, this is a fairly small part of my group, but we are working on it for quite some time. This was in 2020, this was 2021, this is in 2023 with a large aperture. We actually have something that's like 1 centimeter is like really an appreciable aperture. But I think that really we are scratching the surface here, that what can be done with this computation and meta-optics. There are a lot of potential applications. And I think this is really starting off a new direction altogether. So thank you.
Thank you very much, Arka. The floor is now open for questions from the committee. Aidan.
So thank you. Great to see you again. This was beautiful, Arka. So I have a lot of questions, but of course, in the interest of time, I'll limit it. So if it's the symbiotic relationship between back-end computing and the hardware, the solution is never unique. And you have an intuition that you are trying to make the MTF or the point spread function engineering kind of like equal for all the spectral channels. At least, you know, intuitively, you're trying to make that. But at the same level, I argue that is that intuition valid as the back-end computing gets faster, cheaper, more accessible for even mobile systems? Where do you see that intuition failing you to create suboptimal solutions in the holistically designed computational imaging systems? I don't say it's going to completely fail, but I'm saying it may not be the ideal. What do you think about that?
Yeah, you're absolutely right. So here the goal was to capture broadband image, putting all the same effort on everything. So I would like to point out a few things here. Like, for example, when you do neural network-based stuff, right, and you are really looking into the, let's say for display application or imaging, that what does a human being like, right? There's a whole human content that we can actually weight. We definitely have to weight different MTF differently. And these were visible, but I mean, I told you thermal, we can go to mid-wave infrared. So mid-wave infrared and all those applications, you probably, depending on like what kind of scene you have, what kind of light source you have, you might want to also weight this MTF engineering differently. And that's kind of what we have here. So this is the code that we developed where you can play with your figure of merit depending on different application. So we don't think there is in one size fits all kind of solution here. This is, and that's why I think it's a fairly exciting to look into where these things can go. There are multiple different directions where it can go. Yeah.
Thanks.
Next is Harry.
I think Andrea.
Thank you for this talk, Arka. What you describe here is a general strategy using metasurface optics together with computation, and the optics is, essentially analog and passive. There's some, maybe you could comment on the loss of information just having to do with the non-unity efficiency of the, passive optics. And this will influence signal to noise and so forth. And where do you see as being if I have many cascadable elements and I have some significant loss of information, then that compromises the overall imaging. On the other hand, of course, it's very advantageous to take advantage of it. So where's the appropriate dividing line and how do you think about that?
Yeah, so I think this is a very good question. I think that's a question that we all need to answer. I can say, like, if you have active optics, you have nonlinearity, you have reconfigurable, you can do a lot more than what we can do. There is no question about it. And I also think that going more than one optics in current fabrication almost always gives suboptimal performance. That's what we are seeing, because each meta optics has some non-idealities, and non-idealities kind of adds up. So you can do a few different things, but overall, I think what I am kind of thinking right now, that either you make this volume optics that's like really kind of monolithic with the 3D printing, maybe MWR, LWIR, but for visible or even kind of near IR, you want to make a single optics, and more optics actually give you worse performance in experiment, even though theoretically it should give you more because you have more degrees of freedom. So that's what we have seen in experiments. So all our work in this is going with the single optics. But now you can ask that, okay, what for imaging particularly, I think the goal will be refractive, meta optics, and computation. I think we need to go to hybrid optics. Especially conformal optics can give you significant size reduction. There is a huge amount of research going on, a lot of manufacturing techniques coming in. I feel that will be a very important research direction if you really want to go to full color imaging. And I kind of one of the main things that I feel that we are at the brink of a point where meta optics can either go and make a real impact or not. I feel that this is the time when you really think that where it can go, you know, and make like I mean meta optics is already there in many ways, right? But can we make a really big impact? And that's the thing that I think I am most interested in.
I think I saw a question from Andrea popping up and then from Lan. I don't know how you want to go in order.
Yeah, I have one small short question for Arka. It's impressive work. So Arka, AI has been widely used in photonics. Can you elaborate on the role of AI in this work?
Yeah, so in this work, actually, we have not done much AI. So this is automatic differentiation technique. There is no AI involved. We have a deep learning surrogate to go from RCWA to initial design, but that particular one, actually, that deep learning can be completely like stupid deep learning network. We just need a differentiable mapping. We don't actually need to be an intelligent mapping. Our AI comes here is in the computational backend. This particular one where we actually have a diffusion neural network based deconvolution technique. Here, what we do is we take the same optics and then we actually have a dual aperture system. We capture one good camera, this meta optics camera, so that we create a lot of data set and then we train it, right? So I do think that the AI has a lot of power, but at the same time, we need to kind of think about how it gets generalized for optical applications. And so I personally think that we did everything with the winner, because winner is completely generalizable. And then we use AI as an add-on to it. So you have to kind of start with something that works better, and then AI can improve it. That's kind of what my philosophy has been.
That's great. Thanks a lot.
Andrea, did you still have a question, or it's been answered before? Otherwise, I think Dragomir has one.
Yes, I would like to ask, I don't know how this...
No, Andrea, go ahead.
Go ahead, Drago, if you want.
No, you go first, Andrea, please. Okay,
sorry. I would like to ask, first of all, congratulations, Arka, great work and great achievements. The paradigm of meta-optics is that we're able to shrink the size of devices. But we also understand more and more over the years that shrinking has a price. Yeah. Typically, you induce actually challenges in creating aberration-free and large bandwidth imaging systems. So I have two questions. So you beautifully show that you can use computation to correct for that. Yeah. But fundamentally, there is a price that somewhere you are paying in the fact that you are trying to shrink. So my question is, when you compare with the conventional optical systems, what is the comparison in the total size of the system, not just the metasurface itself? And second, would some of these approaches, computational approaches, help also conventional systems? And when you do the comparison, do you still get an improvement in compactness, in compactifying the system itself? Or there is something fundamental of metasurfaces that is helping us just because we're engineering better the overall response? Thank you.
Yeah, very good question. Okay, so there are multiple questions here. Let me kind of maybe give you a short answer here. Meta optics altogether here, you still are limited by the focal length, but we can make short focal length, so you can get some reduction. Once you start doing conformal meta optics, you can show you can actually reduce the total track length because you can actually get a fairly large telephoto ratio. So in some way, replacing 8 lanes with one surface, that is probably not going to happen. But replacing these 8-9 lanes with the conformal meta optics, maybe 2-3 surface, can give you much significant reduction, right? So that's one thing. The second thing that you're saying, I feel that meta optics can solve for spherical aberrations. That's kind of, I think in zero field, meta optics will always be worse than refractive. Larger field angle, it can be better. So can we optimize this with the refractive and meta optics? But frankly, I will say that this is, I am telling you as a success story, to me, this is a failure story because this is probably as good as it gets. And is it good enough? And if it is not good enough, then what we can do? But my whole point here is that this idea that we are solving some of these limitations using computation and the fact that it can be done, that should tell us that there are other problems which we can do better using meta optics and computational imaging. I think that's really what I feel that the main takeaway from my research, that there are computation can help you. And I will say that there are fundamental limits. Computation cannot beat the fundamental limit, but computation can help you to reach that fundamental limit without making like some complicated structure. For example, you can see ours are just simple square. We are not doing dispersion engineering, which can allow us to make in 1 centimeter. You can probably get that with a very, very like maybe nanometer scale precision fabrication, but that's just not possible, right? So computation, and lastly, computation is everywhere. It will be not wise to not take advantage of the computation, right? So that I think is the main bigger message here from my side.
But just to clarify, in this specific slide, when you look at the refractive approach and when you look at the metasurface approach, is it fair to say that the overall thickness of the system is comparable?
So overall thickness of our system is around 10% less, but that's really coming from the optics and this. But this is also not very fair because someone can actually make this lens also, the plastic and everything. So I don't think there is, ultimately you are limited by focal length in this particular case.
Okay.
Yeah.
Thank you.
Thank you very much, Arka and committee, for the nice discussion. We can now move on with the next speaker. We now have the presentation from Gili Bisker from Tel Aviv University, and she will present it to us about advanced optical materials for sensing and imaging in the near infrared. So the floor is yours, Gili.
Sorry, can you hear me now?
Yes, we can hear you and we can see your screen.
Thank you very much. So thank you very much for this opportunity. I'm very excited. My name is Gili Bisker. I'm from Tel Aviv University, and I will tell you today about advanced optical materials for sensing and imaging in the near infrared. So I want to start by acknowledging my group that made it all possible, and of course, the funding sources. So we focus in my lab on near-infrared fluorescent single-wall carbon nanotubes for imaging and sensing applications. There is a large diversity of research directions in the lab. I will briefly go over them. So we work on imaging in the near-infrared, super-resolution imaging of single-wall carbon nanotubes in the near-infrared. We develop nanosensors for bioanalytes. We monitor active processes such as self-assembly and enzymatic activity. We do all this with single-wall carbon nanotubes. So single-wall carbon nanotubes can be thought of as a graphene sheet rolled onto itself to form a cylinder. Depending on the angle of rotation, as you can see in this clip, you get different chiralities. Here you see images of 3 different chiralities. The semiconducting nanotubes can emit fluorescence. And here you can see a typical excitation emission map. The y-axis is the excitation wavelength, and the x-axis is the emission wavelength, and the color codes for intensity. You can see that the emission is within the near-infrared range between 900 to 1400. Here you can see different peaks. Each peak corresponds to a different chirality. The advantage of the fluorescence of the nanotubes is that the fluorescence is non-photobleaching, so you can excite them for a long period of time. And the fluorescence range is within the biological transparency window, where, for example, blood and water and biological samples in general do not absorb much. So these are perfect nanoparticles to work with for biomedical applications. So how do we render them sensors? We functionalize them. We take the single-wall carbon nanotubes, we wrap them or functionalize them with tetrapolymers, which has hydrophobic and hydrophilic parts. These polymers form a complicated corona phase around the nanoparticle. And then if we tailor this functionalization, we can basically get a synthetic molecular recognition agent that target analytes can bind onto its surface. Or we can also induce changes or modulations to this polymer coating. And both of these events, either a binding event or a modulation of the corona phase, is then translated to a change in the emission spectra, which can be manifested in either an intensity change or a wavelength shift. And this is the signal that we detect. For example, this is a functionalized nanotube with functionalization that is sensitive to dopamine. Dopamine is a small molecule neurotransmitter. And upon the interaction with dopamine, the intensity increases. Here you can see nanotubes deposited on a surface, imaged with a two-dimensional camera. When we add dopamine to the sample, the nanotubes light up. And as we wash the dopamine away, the fluorescence intensity returns to baseline. And then we can repeat this experiment again and again and again, getting spatial temporal information about dopamine. Okay, I will start by talking about imaging in the near infrared. This is a D from my lab. So we work with C. elegans worms. They are very popular model organisms for biologic research. And this is just a wild type worm. We didn't do anything to the worm, and we just imaged it under a fluorescent microscope. This is the Ducky channel. You can see here the wavelength. And as you can see, the worm lights up under fluorescent microscopy. This is the overlay. So basically, the tissue of the worm itself emits fluorescence in the visible range. This is throughout the range. This is a GFP, M-Cherry, and Sci5 channels. Okay, so you can see a scheme describing the fluorescence. You can see that the autofluorescence of the worm dies off at about 800 nanometers. So if you look above 900 nanometers, it's completely dark. There's no background. And this is perfect because this is exactly where the nanotube fluoresces. So you have a complete spectral separation between the autofluorescence of the organism and the fluorescence of our nanoparticles. So what we do, we give the nanotubes as food to the worms. They basically eat the nanotubes, and they end up in the lumen of the intestine. You can see here the near-infrared fluorescence of our nanotubes coming from the intestine of the worm, and you can see the huge autofluorescence background. Because we have spectral separation, if we see signal in the near-infrared, it's 100% coming from our particles. So these particles can be used to monitor, for example, the gastrointestinal motility in C. elegans, using the near-infrared fluorescence. And of course, we can quantify it and extract biologically relevant parameters of the contraction of the GI tract of the worm. Okay, now I want to talk about nanosensors for bioanalytes. This is Ronnie from my lab. Ronnie developed a sensor for insulin. She has nanotubes functionalized with pegylated lipids that upon the interaction with insulin, there is a decrease in the fluorescence intensity. This is how the raw data looks like. Fluorescence versus wavelength. The top curve is the initial fluorescence. As we increase the concentration of insulin, there is a gradual decrease in the intensity. Based on such data, we can calibrate the sensor. When we plot the normalized fluorescent response versus the insulin concentration, we can fit it with a hill curve, and then we can provide predictions. Given the response, we can tell what was the concentration. So in order to assess the function of beta cells, this is the cells in our pancreas that are responsible to secrete insulin in response to glucose, we conducted the following experiment. We cultured the beta cells, we added glucose, waited 2 hours for the cells to secrete insulin, collected the conditioned media, mixed with our sensors, and immediately we get the fluorescent response from which we can deduce the concentration. And this is the result that we got. In order to validate our result, we perform ELISA. This is an antibody-based test. And you can see we hit spot on. Of course, we exchange ELISA test, which is a very long test. It has multiple steps. It takes a few hours, with an instant feedback with our sensors. We can also monitor enzymatic activity. This is done from my group, and we were aiming to detect the activity and inhibition of cholinesterase enzymes. So acetylcholine esterase hydrolyzes acetylthiocholine, this is this molecule, to acetic acid and thiocholine. Thiocholine is then recognized by DNA functionalized single-wall carbon nanotubes, resulting in an increase in the fluorescence intensity. Given this mechanism, if there is an inhibition to the cholinesterase activity, then we would see no fluorescent response. I want to emphasize that high doses of inhibition of cholinesterase enzyme is lethal. Low doses of inhibition of cholinesterase can be also used as therapeutics. For example, neostigmine, it is a cholinesterase inhibitor used for treatment of some chronic disease. And if we have this inhibitor, there's no fluorescence change, as you can see here. This is the normalized fluorescent response for two different of our sensors, and you can see that without the inhibitor, the fluorescent response was more than 3 times larger than in the presence of cholinesterase inhibitor. Finally, because these are nanosensors with optical signal, we can have both spatial and temporal resolution to show that we deposited our nanosensors on a surface with the enzyme and the substrate, and then we can follow with time the lighting up of nanotubes throughout the field of view, which provide real-time information and spatial information about the hydrolysis of the molecules in real time. Okay, this is the initial and final snapshots. Okay, we can also monitor self-assembly with our sensors. For example, we work with FMOC tyrosine. The enzymatic oxidation of FMOC tyrosine results in the self-assembly of functionalized melanin-like material. Melanin, by the way, is a pigment, and it protects against UV radiation, and it can also bind metal ions. What we did is we functionalized the nanotubes with FMOC tyrosine and induced the enzymatic oxidation with tyrosinase on the nanotube surface, forming a melanin-like coating of the nanotubes. And then upon the interaction with metals, we can also get fluorescent response to the metal binding. This is how the oxidative polymerization of FMOC tyrosine looks like without the nanotubes. You can see the brownish color typical of melanin, but it also happens in the presence of carbon nanotubes, as you can see here. We can also track this enzymatic oxidation and the self-assembly of melanin from the near-infrared fluorescence, as you can see here. As the oxidation takes place, there is a decrease in the fluorescence intensity. And if you plot both the increase in the absorption and decrease in the fluorescence, we can see that there is perfect correlation between these two processes. And with this melanin functionalization, we can also detect metals. So here you can see the fluorescent response for increasing concentration of iron or for increasing concentration of copper. And we've also managed to get calibration curve for a panel of 9 different metals, all with the melanin-like functionalization around nanotube. By the way, the KD values were in the lower micromolar range, and the limit of detection was in the nanomolar range. Super resolution imaging of single carbon nanotubes. Okay, this is Ronnie from my lab. So what we did is we used an algorithm called super resolution radial fluctuations that was developed in 2016, not by us, for super resolving visible fluorescence images. We adopted this algorithm, and to the best of my knowledge, we were the first to apply it to the near infrared, which is where the challenge is more difficult because of the longer wavelength. And here you can see the before and after of images of nanotubes with low, medium, and high densities. On the right, you see the improvement in the full width of max of these cross sections. Using this algorithm, we can also differentiate between 2 adjacent nanotube, which we cannot differentiate before. And we can also super resolve videos of single carbon nanotubes in solution. Using these results, we then adopted a deep learning approach with a convolutional neural network architecture with the surf images used as the ground truth to super resolve images in real time in compared to the surf algorithm, which is not in real time. Using the deep learning approach, we could improve the resolution of nanotubes with higher temporal resolution. We could remove the edge effect, as you can see here, and we could remove the background noise compared to the surf results. So to summarize, I hope that I've convinced you that single-wall carbon nanotubes are promising optical probes in the near infrared with multiple advantages. We could use them for imaging in vivo in the near infrared biological transparency window in microscopic organisms to track active processes such as the GI contraction. We can monitor supramolecular self-assembly processes like the melanin polymerization and self-assembly on the nanotube surface. And we can also sense target analytes and enzymatic activity with real-time feedback and spatial temporal information. With that, I would like to thank you for your attention and thank my group again. Thank you very much.
Thank you very much, Gili. The floor is open for question. Aidan.
Thank you. This was beautiful. So I have also quite a bit of questions very relevant to some of the things that I do. I'm very excited to see these results. But perhaps to start with, for the super resolution part, I have two questions. First, how do you regulate hallucinations? So what protects your approach from hallucinating, especially which is a known problem of deep learning based super resolution approaches, of course, unless you do something about it. Otherwise, they're very nice to hallucinate. So what are you doing to protect your method from hallucination? And what is the physical origin of super resolution here?
Okay, thank you very much for the question. So the super resolution first, the algorithm is completely, everything is we are training everything on a data set, on labeled data sets, okay? So the ground truth is coming from the surf algorithm, and then it's completely algorithmic approach. I can go back. Okay, so this approach is completely computational, okay? Here, there's no deep learning. We give each pixel or even subpixel a radiality score based on how the gradients of the fluorescence intensity look like. And if it's a radial gradient, then the pixel gets a high score of higher probability of having a fluorophore in this specific location. So this is purely computational, so there's no hallucination in this approach. And then we take the results of the surf algorithm and use that as the ground truth to train our deep neural network algorithms. And frankly, we did not see any hallucinations given the data that we have. And we could also see improvement compared to the result of the surf algorithm.
Where is the information coming from then? So how do you beat the ground truth first? Second, hallucination is always a concern of what kinds of samples you train it with and what kinds of samples you test it with. How do you ensure generalization to various different new types of samples never seen in your training? How do you ensure that hallucination-free operation? And how do you beat the ground truth? Where is the free information coming from?
Oh, okay, sorry. So just to be clear, the surf algorithm is our ground truth. So it's not, we did not try to beat the ground truth. We try to make the algorithm in real time. So the surf algorithm does not give the result in real time. This is why we used it as we used it only for training. And then after the training, the deep neural network approach allowed us to get real-time results. So again, the approach was not to beat the surf algorithm, but rather use it for training, use it as the ground truth, and then improve the temporal performance of the network. And again, the SURF algorithm, it's purely computational, okay? So based on the positions of the fluorophore and the spatial distributions of the fluorescence intensity, from a computational approach, it assess what is the probability of having a fluorophore in each particular pixel.
So regarding new types of samples never seen before, new types of, how did you ensure its generalization? So how did you test the generalization of what you're having?
So we prepared all the samples we prepared ourselves. So there's no synthetic data at all. We developed a protocol to take many images of nanotubes, but of course, you know, it's not that we had thousands of images. Okay, we had approximately hundreds of images, and we showed that this approach works for our for a sample set. If there is a completely different approach to take new data, then all the algorithm is completely free, available online. And if someone needs to train it on their own data set, then they could do that. So we only tested it for our data sets. We took it in the most general way possible. And of course, we had a test set of images, and we performed, and our algorithm performed very well on the test set.
Thank you.
More questions from the committee? Yes, Lan, go ahead.
Hi, great. Thanks a lot for the great talk. Can you explain what is advantages of this single wall fluorescent single wall carbon nanotube compared with other fluorophore for imaging?
Yeah, sure. Thank you very much for this question. So there are several advantages of using single wall carbon nanotubes. So first, between 900 and 1400 nanometers, which is less common than visible fluorophores. There's no autofluorescence, okay? So I can keep, you know, an experiment running for days, weeks, months, and so on, even more than a year, and the nanotubes will still fluoresce. I don't know of any fluorescent dye that is capable of this thing. They're biocompatible, okay? You've seen, we've worked in vivo. We're also working with, I didn't have time to show all the data. We're also working with live cells, and there's no toxicity at all. This is basically an all carbon particle with a biocompatible wrapping around it. So we also get the biocompatibility as a bonus, and we also get the responsiveness to the environment, to the near proximity of the nanotube. So it means that if there is a target analyte that is binding on the surface, or something changes, like self-assembly process happening, an enzymatic activity taking place, then it leaves a mark on the fluorescence emission, and we can then use it as a proxy to tell what is going on the surface of the nanotube. So this combination of properties render them very unique fluorescence nanoparticle for many biomedical applications.
Yeah, thanks a lot for your information.
Thank you.
I think, Aidan, you have another question?
Why don't we go with Dr. Neshev, because I already asked. If there's more time, I'll come back after Dr. Neshev.
Thank you.
Sure.
So I have a very quick question on the functionalization. And can you comment on the binding? Is it a strong binding, or it's a weak binding, and kind of a little bit more on these properties?
Yeah, so thank you very much for the question. So the binding is first non-covalent. If there is an aromatic moiety on the suspending agent, then usually the interaction is pi-pi stacking between the aromatic ring and the graphene lattice of the nanotube surface. You can also have hydrophobic interaction, again, depending on the type of wrapping. If it's a polymer like a DNA, it's very stable. Okay, so the DNA will not come off. Small molecule surfactants, if you dilute the system, then these molecules can come off. You have to be above the CMC, the critical micelle concentration of these small surfactants in order to have high colloidal stability and a stable suspension. So it depends on the type of wrapping, and this is what we do in the lab. The functionalization, this is how we render the nanotubes, applicable for different needs.
Very interesting. Thank you so much.
If you have time for a quick question.
We have. Go ahead.
All right. So in terms of batch-to-batch variation, can you give us some numbers? Like if I were to repeat the same sensor development, what kind of a batch-to-batch variation do you observe? Are these highly repeatable sensors that, if I were to repeat it 1,000 times, for example, with different batches within a year, would you get similar responses?
Okay, so thank you very much for the question. So the answer is yeah, it is very repeatable. This is how we choose our sensors. I wanna bring one example. So if you, again, I didn't show the data here because I didn't have time, but for example, for the insulin sensor, we tried different approaches to find the best insulin sensor, and specifically one of the things that we looked at is reproducibility. We had two approaches, and the second approach we neglected it because it did not show high reproducibility. We made a new batch and the response was different. This is in contrast to the functionalized pegylated lipid nanoparticles, single-wall carbon nanotubes that showed high reproducibility, and every batch that we made showed the same response. So this is definitely something that we consider and take into account.
How do you store these sensors? Like, do you have a shelf lifetime? So do you have any degradation?
So we store them usually in the fridge, but I know firsthand that I store them also on the shelf at room temperature, and they're stable. Perhaps after a long period of time, you would like to just centrifuge again, make sure that, if any aggregates are forming, you can just get rid of them. But basically, the colloidal stability of these suspensions is very high. Again, so I've kept samples for more than a year on the shelf.
Thank you.
Thank you very much, Gili, and the committee for the questions. So we move now on to the conclusive talk of today, which will be given by Junjie Yao from Duke University. So you can share your screen, and then you will be talking to us about the breaking limits in photoacoustic imaging, faster, deeper, and more colorful. So the floor is yours.
Okay, thank you very much. Hope everybody can hear me. Good morning and good evening, everyone. My name is Junjie Yao. I'm an associate professor at Duke. I want to congratulate you all the previous speakers for their wonderful work, and thank the committee for inviting me here. Today, I want to present to you our recent efforts on photoacoustic imaging, and also how this technology breakthrough can enable new discovery, new science, and potentially clinical impact. First, I want to thank my collaborator, my funding agency, for supporting this work, and also my lovely lab at Duke. As you can see, the Duke Chapel in the background, and that's where we live. And so we are at North Carolina. If you haven't been here, you know that we have a lot of trees, and we have a lot of time to go hiking and barbecue. And we use the chapel for lab member weddings as well. And we have a lot of cats, cat lovers. When we do not play with cat, we go to South America or Africa to study frogs. And we also study local frogs as well. We have a very, very strong soccer team in the lab. So if you want to challenge us, you're welcome to let me know. Back to science. If you work with fluorescence, you probably know that fluorescence is from photon to photon conversion, no matter 1 photon or 2 photon fluorescence. For photoacoustic imaging, we actually use photoacoustic effect, which converts the photon energy into sound. By doing so, we can enjoy both the optical contrast of the optical absorption and also the ultrasound provided the resolution and penetration depth, which is not available by other optical imaging modalities. So usually we start with a short laser pulse excitation, the temperature rises tiny, and we get ultrasound pressure, which is detected outside the tissue, and we can reconstruct where the light is absorbed, and so we can recover the optical absorption contrast. While the signal is carried out by ultrasounds, we can enjoy the ultrasound penetration and the resolution. So if you put a photoacoustic imaging into the context context of other optical imaging modality using light, you can see we stand out in terms of its penetration depths with high resolution and also the scalability, because we can configure both light and sound to achieve different resolution and different penetration depths. So what we can image, we love to image anything that absorbs light, right? Either endogenous or exogenous. And for example, hemoglobin that is very abundant in blood, we use that to image the blood vessels very well. And among all the wavelengths, we love to use the near infrared the most because the light goes into the tissue the best. You know, again, the Duke Chapel, we love the blue sky very well, but the blue light doesn't go through tissue that much, while red light or near infrared light can go much better. That's why we love to use the near infrared window the most. And in addition to the endogenous contrast, we can also do exogenous contrast. For example, organic dyes, nanoparticles, or genetically encoded gene products as long as they absorb light. So this is just some basic background about photoacoustic imaging. Then the question is, why photoacoustic imaging, right? What does photoacoustic imaging can provide to the table that can make a difference? I'm also inspired by the first microscope invented by Hooke, which, 100 years ago. I don't think the impact is about the invention itself. The impact of the microscope is the discovery and new science enabled by the microscope. So, himself also discovered cells for the first time. So the same standard should be applied to photoacoustic imaging as well, which this should be evaluated by what kind of new discovery, new science, and new clinical possibility that is enabled by photoacoustic imaging, while other optical imaging or imaging technology may not be able to provide. So that's my thinking, and I actually refer this question to the most intelligent person on planet Earth right now, and I got a very, very positive feedback by ChatGPT. So we're good to go. So at Duke, we are very busy with developing photoacoustic imaging. We focus on 3 research directions, try to break the limits in the technology, make the imaging faster, deeper, and more colorful for molecular imaging. And while we're very busy with the technical development, we have our eyes firmly focused on what can be potentially enabled down the road, what new information we can get enabled by those technical breakthroughs. Here, I'm going to present you a few examples that have demonstrated such potential from technology to discovery. So the first technology is high-speed photoacoustic microscopy we developed with micrometer resolution, centimeter field of view, and kilohertz frame rates that enable us to study dynamics. And this technology is enabled by a few innovations, for example, polygon scanning, wide field ultrasound detection, or Raman-based optical excitation. So the speed actually is more than 1,000 times faster than the traditional photoacoustic microscopy. So we'll see what this high speed can bring us for new science. The first example is that we studied how the epinephrine actually changes the brain functions. The epinephrine is widely used for doctors to treat cardiac arrest, but nobody knows what really happens to the brain when the drug is used. So we studied the mouse brain response to the epinephrine, and we can see very clearly the drug induced a macrovascular constriction and a very persistent hypoxia inside the brain. So which is not a really welcome sign for the patients, right? So this work has been done in the last few years, and the work was recently published and highlighted by editor. Hopefully will provide more insight about this drug in the clinical practice to treat the cardiac patients. The second example is we can use is high-speed imaging to combat the motion. So if you want to image, for example, one of the most challenging targets, the placenta, which is really prone to motion, so it's so close to the belly. And if the image speed is so slow, you will have a lot of artifacts. So we use this high-speed photoacoustic microscopy to study the placenta from the beginning to the end. So we can analyze the macrovessels, their dynamics, their change over the entire pregnancy, both for healthy pregnancy and for pregnancy in disease. For example, preeclampsia, preterm births, or even alcohol abuse. So this is an example that we can combat the motion while to keep the high resolution, high-speed image quality. And the last example is we recently applied photoacoustic imaging to study a very magic animal, the glass frog. We know this frog can become more transparent when they are sleeping, but we just don't know how they do that, right? So this work has been highlighted recently on a Science cover article. We use photoacoustic imaging to study the frog when they are sleeping and when they are active. So we can clearly see that the frog concentrates more than 90% of the red blood cells into the liver, so they can automatically become 4 or 5 times more transparent when they're sleeping. And if you look at the dynamics, you can see how quickly they can remove the red blood cells from the circulation system and head them into the liver to become more transparent. I can tell you that it's not very simple or easy to stay alive in the jungles, right? So this work has been highlighted by many news, and we're very happy to say that because of this new discovery, there are a lot of following interest in this research direction enabled by photoacoustic imaging. So the second example technology I'm presenting you is to break the penetration depths for photoacoustic imaging. Photoacoustic imaging is the same as all the other imaging that use light. You know, if you just want to shoot light from the surface, that's just a fundamental limit of how deep the light can go. Because we're using ultrasound for detection, we don't really have to be limited by that. So we're thinking, can we just deliver light from inside? Just use a catheter or optic fiber deliver light so close to the target deep inside the tissue while we still detect ultrasound from the outside. And then we can achieve much better penetration traditionally achievable by light. In this case, we can go to more than 10 centimeter beneath the skin surface while we still enjoy all the benefits of photoacoustic imaging, functional molecular sensitivity, and we can achieve clinically relevant depths. So the first example by this technology is to study or to monitor the hemorrhage inside the kidney when patient is treated for kidney stone by shock wave. In this case, we deliver light through a ureteroscope. The fiber goes all the way through the channel of the scope all the way to the kidney, and we still detect ultrasound signal from outside the tissue, which is about 5 to 10 centimeter to the skin surface, and then we can monitor what happens when the treatment is ongoing. So we try this on the animal model, pig model, and we can clearly see by the ultrasound there is a optic fiber actually parked inside the kidney, and we can have a glowing kidney, in this case for photoacoustic imaging, deep inside the tissue. And now we can see how the shock wave can damage the blood vessels while we're treating the kidney. And the second example application is clinically relevant is we apply the similar concept for intravascular application. In this case, we can deliver light through optic fiber through the intravascular catheter, and we can get so close to the blood clot for deep vein thrombosis, which is typically 5 to 10 centimeter deep inside the skin, under skin surface. And it's not available for photoacoustic imaging traditionally. So by this case, we can get so close to the blood clot while the ultrasound signal is still detected outside the tissue, and we can provide real-time treatment guidance for the clot. And also we can analyze the chemical composition of the clot. And for example, it's RBC content or fat content. So we can provide the, for example, age information for optimized treatment setting. In this example, we can see a blood clot, which is about 12 centimeter deep underneath the skin surface, and we can analyze what is the oxygenation level of the clot, which is basically, it highly correlates with the age of the clot. And we can also look at how does the treatment change the clot morphology and its chemical composition. So this is 2 examples showing that if we can break the penetration limit of photoacoustic imaging, we can achieve clinical impact by applying this technology to look at specific applications in the patient care. So this work has recently been highlighted by IEEE BME. And in addition to imaging for using ultrasound, we deviated that a little bit recently for new work that using ultrasound for bioprinting. So, ultrasound can go much deeper than light. So we developed a ultrasound sensitive bioink, which can change its phase from liquid to solid when they see the ultrasound pressure. And by doing that, we're able to achieve through tissue bioprinting, centimeter tissue in thickness, and we can get really complex structure printed inside the tissue or inside through the tissue. And for example, for minimal invasive medicine applications. So this example is showing that how we can use this bioink and ultrasound to print a structure to basically fill a hole in a heart. And this is the video showing how this bioink is liquid and it's turned into solid while we apply the ultrasound. So the tissue is open for visualization purpose, but you don't have to do that for the real surgery. And this is just an example that this ultrasound enabled bioprinting can potentially be used for through tissue inside the body surgery purpose. Okay, the last example I'm presenting you is for photoacoustic molecular imaging. And this work has a long history. And in traditional photoacoustic molecular imaging, it always suffers from the contrast issue because the background signal is so strong from blood, which is everywhere. And the other contrast is so weak that we do not see them very well. In the last few years, we have been trying a new strategy for photoacoustic molecular imaging. It's called photo switching photoacoustic imaging, which uses a photo switch of phytochromes. It's a protein that can be freely expressed by cells or tissues. And this protein has two colors. It can be switched back and forth. So by doing that, we're able to, for example, look at a switching tumor which expresses a protein while the background from blood, although it's very strong, but it cannot switch. So we can actually extract the switching signal and have a much, much better sensitivity for detecting those molecular probes, which is more than 1,000 times actually better than the traditional photoacoustic imaging relies on the non-switching mechanism. And recently I've been also working on animal models. We try to make this even more flexible for cell expression or tissue type specific expression. And we have developed a photo switching mouse. Basically, you can freely express the phytochromes at certain organs or tissues or cell types as needed. So that's a much more flexible channel or platform for us to apply this. So two examples enabled by this molecular imaging capability. First, now we can look at the liver, which is traditionally really challenging to image by photoacoustic imaging because the liver is filled with blood. And now we can look at the liver regeneration over 2 weeks. So you can cut part of the liver out. You can see how the blood vessels regenerated pretty well. But now you can also see the liver expression of phytochromes. They can be imaged very well as well because now the sensitivity is much improved. So this is the first time we can look at the liver itself over the 2-week regeneration process. The next example is neurons. We always want to image neurons, but because the brain is such a blood-rich organ, it's always shadowing the other molecular probes. So with this photo switching strategy, now we can see a very specific label neurons in the hippocampus. This is not the max limit we can achieve. We can go through all over the brain, but the contrast or sensitivity is much more improved because of the photo switching capability. So this is 2 examples. I just want trying to show that this new molecular imaging strategy can enable us to image the molecular or neural activities with much better sensitivity that is not available before by traditional photoacoustic molecular imaging. In conclusion, I hope I have presented you today that photoacoustic imaging is a really powerful technology that's intrinsically sensitive to functional and molecular information, which may not be shared by the pure ultrasound imaging as well. And also we are at Duke very busy with developing these technologies that can break the speed, penetration, and sensitivity limit set by traditional photoacoustic imaging. And enabled by those technical breakthroughs, we have already contributed to new science and discoveries, which is not available by probably by other imaging modalities. And we also have our eyes on the clinical translation that's enabled by the deep photoacoustic imaging capability. And with that, I want to thank everyone again for this great opportunity. And I welcome the questions from the audience and the committee. Thank you very much.
Thank you very much, Junjie. So the floor is open for question, and I see Aidan has already his hands raised.
Go ahead.
Nice presentation. This was so fun, and thank you for making it kind of like easier to follow and fun because this is the last talk, and certainly we needed that to bring us back. So I was very intrigued by your Sona ink. I have a few questions on it.
Yeah, go ahead.
First of all, can you tell us more about your lab's contributions on Sona ink? Is it driven by you or by your collaborators? Just for the sake of understanding the interdisciplinary collaboration that you have on it. But more importantly, which is the scientific side, how do you make Sona ink specifically target the features of interest that you have without creating background noise for you? And
so, yeah, go ahead.
And can it be cleared by the kidney? So is this something that you can have within a day or so clear through the body?
Very good questions. Thank you very much. This is I can say this is a fresh work just to get accepted. And This has been 3 years of collaboration between us and a group at Harvard Medical School, Professor Sheng, which is also my graduate program classmate at WashU. So the contribution from our lab is the concept of ultrasound printing. So, and the contribution from my collaborator Shrek's lab is the bioink itself. So we are more on the printing technology and how to optimize and develop a system to achieve fast, efficient, and high resolution printing through tissue. And also all the modeling work that can enable the optimize the printing setting. And Shrek's work is to come up with a brilliant idea how to make a bioink that is biocompatible, as you asked for the second question. And so it's safe for the tissue and safe for the potential clinical application. And I think your third question is maybe two and third question together is how do we get the ink in, how do we get the ink out, right? So
well, more than that, how do you make sure that it's not creating noise for you?
When you say noise, you mean the background printing?
Yes,
right?
yes,
okay.
yes.
So
non-specific, yeah.
Yeah, that's a good question. So right now the ink is not targeted. It's just like a ink you inject into the organ that it's a liquid to begin with. Unless it says ultrasound, it's just a liquid, right? When you apply ultrasound to that, it's due to the ultrasound thermal effect, it will become solid very quickly within sub-seconds. And because of that, the resting or the remaining ink, which is not printed, which is not the same in the ultrasound. In this case, we actually focus ultrasound for spatial confinement. And the ink which is not printed, they're still liquid. And right now what we do is we just use a needle to suck it out. So basically the rest of the liquid phase ink is removed by a needle. But down the road, we're thinking to have a biodegradable bioink that can actually just naturally be degraded by the tissue, which is, we're not there yet. It's just for the future work.
But is it then fair to say that this is a chicken and egg problem? For those objects that you cannot image, you cannot 3D print. And it means you cannot use the Sona ink as a contrast agent for things that you couldn't image well anyways. Is that fair?
So we do not, in this case, we do not do the imaging. The imaging is not, is possible by, for example, using ultrasound as well, because the ultrasound is sensitive to this phase change. But we're not doing ultrasound imaging as right now because right now we use still ex vivo tissue printing. It's not in vivo yet. But I guess down the road it can be combined with a real-time ultrasound or photoacoustic imaging that can provide us with a real-time progress of the printing. Because the different good news is that the color of the bioink doesn't matter. So we can make it really photoacoustic imaging friendly. And when it's printed, when the phase changes, you can also change its color. So we can use photoacoustic imaging to see the color change to give us a guidance of the printing process, which part is printed, which part is not printed. So that's a future plan we're thinking of.
Thanks.
Thank you. Very good question.
Are there other questions from the committee?
While people are thinking, I might. So you mentioned you had a breakthrough in depth in photoacoustic imaging, but maybe I'm tired. It's been a long morning for us. What was the scientific ingredient for that breakthrough? Can you tell us? Yeah.
Good. So I have to skip all the details. I'm sorry about that. But the engineering part is really challenging for us because we have to deliver sufficient light into the tissue, which can provide efficient illumination of deep organs. Can still be detected by the outside ultrasound transducer. So the concept of separating light and sound, which is actually pretty new for photoacoustic imaging. Traditionally, we always do them together. So light and sound stay together. The light source and the sound detection are together. That limits how much we can put into a catheter, for example. So now we separate them. The light can be delivered freely by a soft, thin optical fiber with our engineered diffuser at the tip, so they can shine a big volume of tissue inside the body, while the ultrasound is intentionally separated on the outside. So we can use all the advanced ultrasound detection technology like array or imaging reconstruction program that can help us improve the resolution and the image quality from the ultrasound perspective. So in this case, we're not limited by the space anymore. So the space is just the catheter or the intravascular catheter is only for light delivery. And the sound is all set. So that can be as fancy as you want. So
basically,
that concept,
it's an architectural improvement, not a scientific breakthrough, you're saying.
Yes, it's engineering.
Well, yeah, okay. Thank you.
Thank you very much.
Thank you very much. If there are no more questions from the committee, we are also coming on time to the end of this part of the event. Maybe there is any last second question. If not, I would like to join me in thanking all the speakers that presented today with these fantastic talks and very engaging and very diverse too. And I would like to ask the committee to send their grades to Junjie and also to Idol within the next 15 minutes while we start the panel discussion inspired by these very nice talks that we just heard. So maybe I would like to ask all the committee members to share a few thoughts or reflections based on these talks that they have heard, maybe on the technological impact, and maybe because it's the rising stars of light, really on the importance of optics and photonics in developing these new technologies that we have seen today. I don't know who would like to start and share some thoughts.
I can start since my mic is on. So I'm very impressed with all the talks. It's been a long day for me, but it kept me engaged, and that was amazing. Very difficult. The winners are, it's just not about winners. It's about celebration of science, photonics and optics science and its impact. And I've seen quite a bit of diversity in terms of both institutions, their locations, types of problems that they're approaching, and the impact that they will generate in society for the taxpayer money. I'm very excited to see this, and I congratulate all the panelists, all the presenters today. I think they should be proud of what they have done, and they are great ambassadors for optics and photonics field.
Thank you, Aidan. Maybe also I would say one thing that was quite exciting is also to see the interdisciplinarity that each and every one of these projects had to accomplish to really build new either technologies or new knowledge at the interface of the material science, the optics, the engineering. So any of you wants to comment a little bit on the importance of this interdisciplinarity these days within the application of photonics?
Yeah. I can comment on something. I'm truly impressed by this cohort of exceptional researchers. They really showcase those are the rising stars that push forward the boundary of science. For example, today I learned about advantages of a carbon nanotube. Why is that as a fluorophore? Because of unique advantages. And also Junjie's talk about photoacoustic imaging and Sang-Hoon base. It's 2D materials that really will push forward heterogeneous integration for innovative photonics. So I think their topics so diverse really demonstrate the multidisciplinary nature of photonics itself. We really need scientists from all disciplines, from material science, electrical engineering, and even computer scientists to work together to push things forward. And so in the end, I wanted to congratulate all the finalists. I think to this stage, it will take a lot of effort. And it's built on the research accomplishment, and the impact of the work will undoubtedly extend and beyond the competition, influencing the future of the photonics world. And thank you once again. Congratulate to all the finalists.
Yeah, I want to echo what has been already said. Thank you. First of all, Junjie and all the organizers to bring us together and put together this amazing event every year since a few years, and every year I feel it gets better. A lot of great talks we've heard today, and the bar is very high. So I congratulate already the speakers, first of all, for the great work, and second, for having made it until now. I agree with Aydogan, the selection would be very difficult. I think everyone is on par, an exceptional young scientist, and I'm glad to have spent these few hours together to hear the progress. We are seeing how much interdisciplinarity, how much photonics is helping technologies in various fronts. We've seen biomedical has been actually a common thread around various talks, but also imaging and computation. It's a very exciting opportunity to be working in this field and to see a new generation coming up. Thank you.
Yes, I can also say a few words. I have been with this event from the very beginning. I think Arden and Andrea and others are in the same team. The quality of the presenters and also the presentations are really great. And I think also I say even tougher job for us to choose the ones to be the finalists. I agree with all the other speakers about the committee members. And also I say the presentations and the work covers many hot research areas in the field. In particular this year, I think I see lots of interdisciplinary research connecting biology, biomedical imaging with how to utilize advanced photonics laser technology. And this is really great. I think I would like really congratulate all the presenters and all the participants. And I really wish even more successful ones in future. Yeah.
Yeah, I think this illustrates that the field of photonics is a really vibrant, healthy, and interdisciplinary field. And I appreciate the fearlessness and the relentlessness with which the speakers are obviously engaged with very difficult and challenging problems in imaging, spectroscopy, and information recovery from really innovative photonics experiments. I think it and it's quite interesting to see over the course of time, as you see this generation emerging, there's a real interest and hunger to have advances in photonics really be immediately applied in an impactful setting. And I think that was clearly in evidence in the presentations today.
Julie, if I may add something, like my observation was from listening to all these fantastic talks was that we're seeing this progression of like really fundamental ideas to some true breakthrough applications in impact to the society, ranging from even clinical applications to new types of imaging devices. So this is absolutely impressive that all the people that we've seen here have really taken the strength and the enthusiasm to walk all these paths, because often people just stop somewhere in the middle and do not have the strength to go and really push these to devices. What are the new devices? What are the new applications that we can do with this breakthrough science? And it's really indeed a great examples of photonics in applications for every impact to the society, from health to imaging and so on. And indeed, it will be a very difficult selection. I think every one of the people who presented today is a winner. We probably only can nominate one, but everybody can be very proud with what they have achieved. And thank you for presenting today.
Thank you for the panel for sharing these comments. I also wanted to ask you, based also on your longstanding experience in the field, how do you see the field evolving forward based also on the examples that you have seen today of these works? Where do you think the translational challenges will lie ahead in bringing these amazing works even further out from the lab and really into the real world? Maybe if you have any thoughts or based also on your experience.
What I think was clear from many of these talks is that, and I think also this is in line with Drago's comment, that beyond the basic science that in which photonics has always thrived, also the engineering aspects. There is more and more importance in making devices that actually work, that compete with other technologies and on various metrics. So I think all the finalists have shown really impressive devices, not just the basic science aspects. At the same time, of course, there is a lot to discover, also from understanding how light interacts with materials. We've seen also a very nice talk on the 2D material aspects as new platform for devices. So I think this is a nice combination. I think as we go forward, there will be more and more of this opportunity to merge engineering with physics and optical sciences.
Yeah, I also want to echo what Andrew just said. I think all of the finalists successfully demonstrated the potential of taking the science into technology that can impact real-world application. And it was noting, for example, it's also beyond that. It's not like a bring to the outside world, but also push the limitation that's set by the current technology. For example, the Arka mentioned that there's 2D materials, this metal surface will shrink these bulky 3D structures into a miniaturized system. And also currently silicon photonics, the silicon materials is not a direct bandgap structure, has a lot of limitation. But if you can integrate these 2D materials with through the heterogeneous integration, it will truly achieve a new kind of silicon photonics with unprecedented functionality and features. So I'm really excited by the talk given today. And I feel really proud of seeing those young generation of scientists writing in the community. They will shape the future of photonics.
I can add a few words. So one common theme is all the way from basic science to applications. We see a large spectrum, and research is becoming more and more interdisciplinary, and the teams are supposed to work together. So we need, first of all, more funding to sustain that. And we also need international teams getting together without any boundaries, without any borders. And I think that's something that we especially need more than ever before, because science, photonics science, optics science, and in general science is a phenomenal ambassador in the world. That brings together various different backgrounds and equalizes them based on scholarship. It's very powerful, it's very important. And every one of these publications that we were seeing through these presentations, by and large, they were coming from various different parts of the world, coming together. And that's phenomenal, that's amazing. But we have to tell this story to everybody that science is actually a great way of bringing people together and helping create bridges among different parts of the world. And I want to emphasize that it's a very important mission of science, and we're seeing live examples of it in every single presentation that we had in the last couple of hours.
Thank you, Aidegan, for this thought. And maybe on this note, we can also ask our rising stars to maybe share some thoughts on their experiences in establishing these fantastic interdisciplinary collaborations, and maybe saying a few words on how did the research found resonance with people or technologists in different fields, and how these amazing works came to be and to the state that we could see today. So maybe each of you, if you can share a few words on the feedback that you got while you were developing these works.
I can say a few words if to start. So I do echo many of your comments about the science and technology right now is not a single lab task. It has to be combined. It has to be integrated over a large team. And for example, the work we're doing with the Sang-Hoon ink, it's based on just a chat over a coffee time with my friend, right? So who's studying chemistry. And so this example has happened again and again in my lab and also in many other teams. And I do feel that open science and the open research communication and also free flowing of knowledge and technology and personnel is very important for us to push every single research front, break the limits of every single research front, and also achieve something that a single lab, a single school, a single country cannot do. That's all.